my welcoming remarks yesterday. So I don't want to repeat that because most of you are here. But just to say that we are delighted that you have been able again to come to today's sessions. And this international conference has been jointly organized with Mount Kenya University and AIBBC. And so what I want to do, if you look at the program, I'll be doing my presentation after these preliminaries. We were actually supposed to start at 8, but we are very much on time because the actual presentation starts at 9. So at this juncture, what I want to do is to call upon the representative of Mount Kenya University, who is representing the Vice Chancellor. Her name is Dr. Marceline Kamande, who is the Director, DVC, Deputy Vice Chancellor, Academic and Research Affairs. So, Dr. Kamande, welcome to give remarks on behalf of Mount Kenya University.
Asante sana. Yeah, as you've been told, my name is Dr. Marceline Kamande, the Deputy Vice Chancellor, Academic and Research Affairs. And I am happy to be here. I was just saying that whenever I come to a scientific conference like this, it always amazes me what we are able to do as professionals and even as in our own individual capacity. So I really want to appreciate the time that has gone into preparing this. And as Mount Kenya University, we are really happy to be partnering with KIPRE and all the other organizations that are involved in this. So my humble duty this morning is to represent my Vice Chancellor, Professor Deogratius Jaganye, in giving a welcoming remark to this function. And so you will allow me just to go straight to that, even as we proceed. Good morning, distinguished delegates, esteemed colleagues, partners, and friends of science. It gives me immense pleasure to welcome you all to the MKU AIBBC KIPRE Joint Conference 2025 here at Olesereni. Just yesterday, we concluded the AIBBC 2025 workshops. And just to note that for the past 3 days, the workshops have been happening at the university. And so we are happy to culminate this week with joining into this conference, which I am informed that it started yesterday with a cross-section session for KIPRE. And now we join into these 2 days just to be able to share what is happening between these two institutions. And so the workshops were an inspiring 3 days that brought together young scientists, postgraduate students and faculty from across the globe. We saw an incredible display of curiosity, technical skill, and commitment to learning, from mastering flow cytometry and molecular diagnostics to exploring genomics, immunology, and parasitic diseases. And I really want to confirm that we had a very big team of scientists that were upcoming scientists, and the interaction between the juniors and the seniors was amazing. So today, as we transition from the workshops to this grad conference, we move from hands-on learning to ideas exchange, from laboratory benches to the stage of global dialogue. And this marks a defining moment where training evolves into thought, leadership, with wisdom of experienced researchers. So this joint conference between Mount Kenya University and the Kenya Institute of Primate Research represents more than an academic partnership. It embodies a shared vision for Africa-led biomedical innovation. Our collaboration demonstrates that when universities, research institutes, and partners unite, we not only strengthen capacity, but also create an environment where science can thrive sustainably. And I wish to sincerely acknowledge and appreciate Dr. Peter Mwethera, the Director General of KIPRE, and his dedicated team for their unwavering partnership and leadership. So thank you so much for choosing Mount Kenya to partner with you in this conference. We are indeed very delighted, and we are happy at where we are going in terms of this partnership. I know if my Vice Chancellor was here, he always tells us when we get into partnerships that we don't want partnerships that take, or rather that gather dust on the shelves. We want partnerships that are able to bear results. And this is one of the partnerships that we are really happy that it is bearing fruit, and there is a lot of interaction. So together, MKU and KIPRE are nurturing a platform that enables research translation, encourages cross-disciplinary engagement, and promotes evidence-based policy development, key ingredients in advancing Africans' health agenda. Colleagues, Africa stands at a turning point in biomedical science. We are witnessing unprecedented investment in research, infrastructure, digital innovation, and scientific networks. Yet, as we celebrate progress, we must remain clear-eyed about our mission to develop homegrown solutions for African health challenges. So through this conference, we explore frontiers such as precision medicine, AI-driven bioinformatics, vaccine development, drug resistance, and climate health interaction. These are not abstract topics. They are central to improving the lives of millions. One of the most encouraging features of this conference is the Young Investigator sessions. These sessions remind us that the future of African science lies in the hands of our youth. Their creativity, courage, and curiosity must be met with mentorship and opportunity. At Mount Kenya University, we have made it a priority to bring research, to build research capacity, training, emerging scientists, and support innovation ecosystem that enable discovery. We are committed to ensuring that our students and collaborators do not just learn science, but they also lead science. I am equally thrilled to note the participation of world-class leaders from the International Vaccine Institute, Science for Africa Foundation, Kansas University, and UPRA University, among others, joining us in person and online. Your engagement underscores that science transcends borders. It reminds us that collaboration between Africa institutions and global scientific community is not about dependency, it is about partnership, mutual respect, and shared growth. Ladies and gentlemen, as we embark on these 2 days of rich discussions, presentations, and networking, I urge us to keep one guiding question in mind. How can our science directly contribute to African well-being? Let us ensure that the knowledge generated here today finds its way into clinics, classrooms, and communities. Let's ensure that partnerships forged here continue to flourish. This conference allow me to express my heartfelt appreciation to AIBBC Organizing Committee, the KIPRE team, our sponsors, and every participant who made this possible. Your dedication and generosity have created a platform that reflects the very best of African science, collaborative, innovative, and forward-looking to all presenters and participants. May this conference spark new ideas, strengthen old friendships, and birth new collaborations that push the frontiers of biomedical research forever. Just in closing, I want to say that something that the Vice Chancellor always says, that when you come to Kenya, please ensure that you enjoy the sceneries of Kenya. For the people who have come from abroad, please enjoy Kenya. And if you can create a moment to come and see where Mount Kenya University is, you are most welcome. We are about 40 kilometers away from here, and you are most welcome. And with that, let us continue to run, to share, and to innovate together for healthier Africa. Thank you.
I would now like to call upon Professor Collins Ouma from African International Biotechnology and Biomedical Consortium to make his remarks and presentation. Welcome, Prof.
Members of the board and advisory committee, KIPRE, our partners from Mount Kenya University and other academic institutions and research institutions, delegates, speakers, good morning. My name is Professor Colin Souma. I'm from Maseno University. I'm the general chair of the IABBC series of conferences. So I want just to join my colleagues in welcoming you to this series of conferences jointly organized by KIPRE and Mount Kenya University and a team of sponsors who made this possible. I want to appreciate all the efforts that have been made in ensuring that this has actually happened. Initially we began, just to take you briefly back in a minute, we started off the conferences initially, and then we thought that our students, postgraduate students and even undergraduate students needed to develop some skills that will enable them to become high-level scientists. So there was a need that was created that actually necessitated that we begin with the workshops, because the students came back to us and told us that just by sharing all those cutting-edge research and results were not really going to help them unless they understood how those things were generated. So that actually pushed us a little bit to start off with the workshops. And then now, traditionally now we do 3 days workshops, and then we end up with 2 days conferences. So we are glad that our students met some of the most renowned scientists within the world within the last 3 days. They shared, they had experiences, and now they have the opportunity today and tomorrow to get to know what those skills that they were actually being taken through within the 3 days translate into research outputs. And that way, we get to now transition them from the bench into the practical world of science that we really wanted them to have. So there's been a lot of debate right now about us as scientists sitting amongst ourselves and sharing just information amongst ourselves without necessarily thinking about the community impact. The current debate right now is how we need to transition the research that we really appreciate within our setup into something that will be impactable into the population of society. So there's been a lot of drive currently in engagement in research to policy to action. So I want to urge our scientists that as we continue to engage within the next 2 days, let's think a little bit about what impact some of these research outputs are going to have within our society. Because if we don't do that, then research just becomes useless. It just sits on the benches. We count publications, and then we also get them for promotions within our academic setups. But let's think a little bit about the society where we come from and how this type of outputs that we are going to share actually drive the impact within society. So as we engage our students, let them also remember that science has become very competitive. One of the things that we nowadays also talk about is collaborative network, because everything has gone multidisciplinary, multi-institutional, and if you don't network and collaborate, then there's nowhere you are going in science. So one of the things that I've always overemphasized that you need to network, collaborate, because that person sitting next to you might just be the person you need to be able to apply for the next grant. So think a little bit about the people you are going to engage as we move forward. As I conclude, because of time, I just want to appreciate the fact that we are all here together, we are here for a common goal, and let's join hands in making sure that we drive the science forward as much as we can. Thank you so much and welcome.
morning, everyone. Again, my name is Dr. Atunga Nyachio, the Director of Research and Product Development at the Kenya Institute of Primate Research. I'm happy to see you this morning, very bright faces, and eagerly waiting for serious presentation this morning. I want to thank Dr. Muidera for the session that has just ended, and also the presentations from the two speakers this morning. We are really delighted, and we are happy for the opening remarks from MKU and also from Professor Collins Ouma. So today, yeah, some question? So we want to go straight to the presentation, and I will request that today we be saving some time. If you see you have been allocated 20 minutes, means you are presenting for 15 minutes, and you allow 5 minutes for comments and feedback. And this morning we have two presenters who are going to give us a keynote presentation. That is Dr. Peter Muidera, the Director General KIPRE. And also we'll have another presentation from Professor Jerome Kim. They have been allocated 30 minutes, so if they can present like 20 minutes and allow some few minutes for discussion, it will be very good. So allow me now welcome Dr. Peter Muidera Gishui. Dr. Peter Muidera Gishui is the Director General at the Kenya Institute of Primate Research, and this morning is going to give a presentation titled Research Towards Universal Health Coverage, a Case for Medical Products, Smooth-Gel, Smooth-Scan, and Unipron. Dr. Muidera is a biomedical scientist with a strong background in reproductive health and the health care industry. He holds degrees from Bristol University, UK, the University of North Carolina at Chapel Hill, USA, and the University of Leuven, Gasthuisberg Hospital, Belgium. His research has led to the development of several innovative products, including two medical gels, Smooth-Gel and Smooth-Scan, which facilitate safe childbirth and are particularly beneficial for women who have undergone female genital mutilation. Dr. Muidera has received numerous awards for his innovations, including the Tony Elumelu Entrepreneurship Award in Africa 2015, the African Union Innovation Award in Brazzaville 2013, and the Best Prize in the Science and Innovation category at the Kenya Public Service Innovation and Award Scheme 2012. He also received the Best Prize at the National Council for Science and Technology Awards in Kenya 2012. In 2019, he was honored with the Head of State Commendation, Moran of the Order of the Burning Spear, MBS, for his contributions to scientific research. Additionally, Dr. Muidera has supervised and mentored several PhD and MSc students and currently serves as an external examiner at the University of Nairobi. Let us welcome him by a round of applause.
Thank you very much, Dr. Atuga, for that very flowery introduction. As I said, my name is Peter Gishui Muidera, but that name was unilaterally changed to Gish by my former supervisor without my consent, but I accepted. And what I'm going to do is basically to summarize the activities that I've carried out over the last 17 years. And it's about reproductive health. And the reason why I chose reproductive health is because this is probably the most neglected aspect of human health. Those of you who remember in 2019, there is a girl who committed suicide in Bomet. And she committed suicide because she started menstruating in class. And she was ridiculed by both boys and girls, including her own class teacher, who was a lady. And she went home and she had a mother and committed suicide, meaning that even the mother did not help. So these are some of the things that make us know that this is a very neglected aspect of human health. So before I do this, before I go into my presentation, I want to start by lamenting that a number of research activities are carried out in Kenya and Africa in general, yet large number of people live in poverty, starvation, poor health, and malnutrition. Few research findings in form of products in the country and Africa add up to the end users, and that's exactly what Professor Collins was referring to, that we need to convert our research into tangible products that can help our people. But this can be leveraged by partnerships between research institutions and industries, because this is how you can narrow the gap between theory and practice, and hence accelerate conversion of knowledge into products and services. But more critically, innovations, technology, entrepreneurship, and manufacturing are going to spur economic growth if we embrace them, and this then will expand opportunities and reduce poverty. I want to give two lessons that we have learned before. In the 1990s, Israel invested 100 billion Kenya shillings into research and innovations, and that created a very vibrant innovation sector in the country. The Singapore story, you all know it, that Kenya attained independence after Singapore because they did get their independence in 1965. We got ours in 1963. But because of emphasizing in research, technology, and innovations, you can see the kind of vibrancy that we have seen in the ecosystem within the innovation sector. But more importantly, we need to think about incubation services. And in this particular case, I would want to refer to two of the incubations that have helped my work as I moved towards the commercialization. And this is Village Growth, abbreviated as VILGROW, and the Tori Emul Foundation. These are two foundations that have incubated my services so that I'm able to commercialize the products. Now, the COVID-19 story, any government that did not learn from the COVID-19 pandemic will never learn anything, because it's only countries that have invested in research, science, and technology that were able to deal with the COVID-19 outbreak. But more importantly, it's good to be wary of products because of this valley of death, because most of the prototypes in the process of our innovations do not see the end of the day because of issues to do with commercializations, issues to do with the funding gaps. So I want to talk about 3 products that we have developed over the last 17 years. Uniplon is a microbicidal contraceptive gel, a gel that has capacity to kill the HIV virus in the vaginal region, at the same time confer contraceptive properties, meaning that it's a strategy that you can give women to empower themselves sexually, because a woman just needs to put that gel in that particular region, and that does not require consent from the man. Smooth gel is a lubricating gel that is already in the Kenyan market. It's already in use. If you go to any chemist or to the hospitals, you will see it as a lubricating gel. It's the only African-developed product of this nature, and it competes with KY Jelly, which you all know that is from Johnson & Johnson. Smooth Scan is a ultrasonic gel for ultrasound. You want to observe the development of a baby in the womb, you smear it on the tummy, and then that allows the gynae to observe the development of the baby using the ultrasound machine. Now, the point I'm trying to drive home here is that when you look at universal health coverage, you'll not be able to roll out serious universal health coverage if you do not have products that are going to be put in the health systems. And that is why we need to start thinking about how do we build capacity within our systems so that we can say that we are able to implement the universal health coverage. This is one of my best slides because all this work that I'm going to present today was carried out in these baboons. And baboons are very close to us in all parameters from physiology to anatomy. But one thing I like about baboons, unlike humans, is that they're less argumentative. So there is an urgent need to develop a product that can give women authority to take control over their sexuality. The reason why sometimes the condoms have not worked is because they are dependent on the men. Because the female condom, when it was introduced into the market, there was an argument that it was making a lot of noise. And because in most of the cases, the women who want to use some of these products, they are at the base, they are charged economically, so they are the whim of the man. So it is important to come up with a product that women can use without consent from the man. It is estimated that a microbicide with only 40% efficacy against HIV transmission, and estimated that a microbicide with only 40% of efficacy against HIV transmission and 30% coverage, so you cover only 30%, can avert infection rates of numbers in terms of numbers of 5.6 million. And 4 million infections if the coverage is at 20%. And this underscores the relevance of a microbicide in terms of HIV infection prevention. So when you look at our country, we have over 42 drug manufacturing companies. And yet we are reliant on imported medicines because Kenya imports more than 70% of all our medicines from abroad. So you can imagine what happens when we have a lockdown like we did during the COVID pandemic. It means our health systems would literally shut down. And that is why it's important for us to think of how we can build capacity in terms of manufacturing our own medical products. So I talked about what Uniplon is, and I'm going to repeat that. We completed the studies in baboons, and we've been able to publish data to show that this is a product that works in terms of HIV infection and also contraception, at least in terms of the baboons. And we've been looking for funds to do human studies so that we can then take these products to another level. Now, Smoothgel and SmoothScan are already in the Kenyan market. After approval by the relevant regulatory authorities that include Pharmacy and Poisons Board and Kenya Bureau of Standards. Our goal is to improve human sexual reproductive health, child and maternal health, and reduce the cost of healthcare and medical care in Kenya and in Africa. The formulation for this Uniplon, Uniplon is a fluffy, acidic, and a detergent lubricating gel that has been carefully formulated in collaboration with a local WHO certified pharmaceutical manufacturing facility called Universal Corporation Limited. The product is heavily buffered with a pH of 3.4 and contains a number of raw materials, and the buffering agent in this product is carbomer. It is in the classification of antifertility microbicidal agent, and the product is very stable at 22 degrees centigrade and also at body temperature. And it does have that patent. And let me say that, as I indicated earlier, when you look at the sub-Saharan Africa, we have a lot of challenges in terms of reproductive health. And there are many contributors to poor levels of reproductive health. One of it is FGM, female genital mutilation. And in that, map of Africa, you can see the levels of FGM. In some countries is as high as close to 90%. So when you look at the total number of women in terms of estimation who have undergone this vice, because this is sexual violence against women, we have approximately 200 million women who have undergone FGM. And this has implications in terms of dryness during sex and complications during childbirth. And it is quite difficult to understand why one would engage in a vice like this that has all these effects on women. So as a team, we developed these products to address these issues. And I don't want to go into the details of that table, but what I want you to appreciate is the fact that for us to be able to reduce maternal death in the country and in Africa, we have to improve maternal care, and we have to improve the access to contraceptives. But for this to happen, we need to provide services for this. So the Smooth is both an over-the-counter product, but also a hospital product. So you can get it over the counter, but you also get it from health institutions. SmoothScan is basically a business-to-business medical product because you can only get it in a facility where there is an ultrasound machine. Now, we target individual clients, chemists, doctors. Kenya Medical Supplies Agency actually are buying this product. Mission for Essential Drug Supplies, this is for the hospitals, mission hospitals, health institutions, governments, women groups, and NGOs, among others. And we reach out to our clients through distribution channels, chemists, agents, wholesalers, KEMSA meds, hospitals, NGOs, and doctors' prescriptions. These products are also aimed at demonstrating that you can reduce the cost of healthcare if you come up with local products that are better priced than the imported products. And this is what this slide basically shows you, that the two products that are in the Kenyan market, they cost less than 50% of the same products, competing products that are imported. We do have a patent for these products. We have other intellectual property instruments like the trade names and trademarks. We have, I don't know why it's not stable. We have done more than 16 publications in peer-reviewed journals. This work has been presented in 17 conference proceedings. It produced one PhD graduate, Dr. Jairo Biero, 2 master's degrees, and other trainings. In terms of the progress to date, the preclinical trials for Unipron was achieved, and for the other 2, the emphasis is now to reach out to the other parts of the African continent. We have won several awards, and I don't want to go through all these, but they're quite a number. In 2013, we won the African Union Innovation Award for these innovations. So I went to Brazzaville to receive the award. And I must say that I was a bit disappointed when I went to Brazzaville for this award because Kenya won 4 awards, and it was #1 in the entire African continent because we won in the health category. The Kenya Defense Forces won in the category of innovations because they developed a mobile kitchen that you can use in combat in the field. The Coffee Research Foundation won an award because they developed a coffee-resistant variety of coffee called Batian. And the veterinary labs won because they developed a digital pen that can connect farmers with information to do with farming. And despite the fact that we were number one in the entire continent, there was no representation from the government. And it is only Kenya, with all that stand performance, that did not have a representative either from the president, prime minister, deputy president, or a minister for health. And I wish the government officers would be here because this I've repeated several times. It's very important that we take research and innovation seriously as a country. And that is why yesterday during my welcoming remarks, I did say that for the first time, this country has dropped below the 100 key innovators in the world. And as a government, we really need to take note of that and take remedial measures. The institutions that have been involved in the development of these products include KIPRE, the Aga Khan Hospital, the Karen Hospital, I don't know why it's flashing. The company that we gave the manufacturing rights are called Universal Corporation Limited, who have been manufacturing these products on contract basis. And this is the only WHO certified pharmaceutical manufacturing facility in this part of the world. And when they set up this facility in Kenya, 45% was by Finnish government FinFAD. But since then, they have sold 51% to one of the biggest pharma in India called Strides. So, what happened? What has happened? But I think I'm coming to the end of the presentations. But just to emphasize that, just like what you've heard from Professor Corin, it is important that we think of how we can convert our ideas into products and services. Let us not do research just for curiosity purposes. Let us ensure that our research activities are commercializable and they're implementable so that even the government can see the need to invest in innovations in research, science, and technology. And I think I only had one more slide, which was basically showing the interconnectivity between all the different sectors, which I would not be so much worried even if it didn't come up. So I'm ready to take questions and to thank you all for listening to me. Thank you very much. I think I've mentioned all the teams that were involved there. We have 2 Guinness in that team. We have Dr. Jael Obiero. I don't know where she is. Jael was basically the researcher on the bench, so we agreed with her that she does the work, but I take the credit. And then we have, as one of the members of the team, the current chairman of Equity Bank, who is also a clinician, Professor Macharia. And then we have a business person to guide us in the business processes. And we also have a local company that is involved in the distribution. Because when you do these things, one of the things you don't want to do is to leave the company that manufactures to also do the distribution because you may not be able to monitor. So we did get a company that is doing the distribution. So we do make the order to the manufacturing company, and then we give the products to the distributing companies. Thank you very much.
Thank you very much, Dr. Muidera, for that presentation. You know, I know Dr. Muidera as a science entrepreneur. So for me, when he speaks, he's speaking business, and that's why he's talking about products. So I'm also trying to learn how to be a businessman, but slowly I will become one. So I think it will be good so that we can take some questions as they are setting up for the next presenter. So we will request him to take a pen and paper, and then we can have some people asking questions or giving some comments, and then I will he will then be able to respond. We have saved some time, and thank you, Dr. for saving us some time. So do we have some questions from the floor? Yes, there are questions from the floor. So I will request, yes.
Can I proceed? Professor Collins is still my name. Yeah, thank you, Doc, for the presentation. On your title page, where you gave the title, you were saying that the gels that you developed actually prevent HIV and then immobilize sperms, if I got you correctly. But then now I'm thinking about those who still want to use the gel. They want to prevent HIV infection and other STIs, but they still want to have children. So is there a thinking process where you can now come up with another gel which can allow them to prevent HIV and other STI, but at the same time allow them to reproduce?
Yeah, that's a beautiful question. One of the things that we did was to understand how reversible this product is in terms of contraception. We did get a system from Belgium for restraining baboons, and from there we were able to apply the gel into the vaginal region and measure the pH levels over a certain interval. And we were able to establish that the product works to a maximum of 3 hours. So in terms of contraception, all what you need to do is to avoid sex within those 3 hours. And I think this is one of the easiest reversibility in terms of time, because you only need to appreciate that it works or is effective within 3 hours. So the pH goes back to normal after 3 hours. There is a question there.
Okay, good morning, Doc. Thank you for the presentation right here. So I'm wondering, can you comment about the process of patenting, how easy it was or how difficult it was? But also, how do we encourage the young investigators, because some of them probably do not know the patenting process, and how do we lobby for support? Because the reason why we are not featuring in the innovation is because people are not applying for patents. Thank you.
If you talk to the people in KIPI, these are the people who are in charge of patenting in our country. They'll tell you the process should be invent, protect, and then commercialize. So if you think that you have a product that can be commercialized, you need to patent it before you commercialize, because the moment you put it in the media or you put it into the public, then there are always busybodies who are going to take advantage and claim it. You all remember the story of the Kyondo. The process for patenting is quite lengthy. One of the things as an institution that we are trying to do is to see how we can facilitate that. Now, of course, the process is also expensive. And that's why we need to strike a balance. Do we want to patent any product that we think that should be protected? Because you can end up having so many products that you have protected, but they never see commercialization stage. And remember, a patent is only valid for 20 years. After 20 years, then it's open to the public. That's why today you can manufacture paracetamol and all those products, and all the vaccines, including the polio vaccines, whose life has gone past 20 years. So the issue here is that there has to be a balance. You have to keep renewing every year. So you can imagine you have a patent that you have renewed for 20 years, you have not commercialized, and then it's been open to the public. But what I think we need to do is to call experts from KIPI and come and give us an assessment of what is patentable in our case. Remember, for a product to be patented, it has to be innovative. It has to be new. You cannot, for example, go and patent a tree, something that is naturally occurring. And also remember that even when you patent in your country, you also need to designate the other countries where you want to patent. So if I feel that my interest is India and China because now that's where the population is high, then what I'll do is that I designate Kenya, and then I also go and designate China and India. In terms of how we can support the upcoming scientists, I know that when I was the chair of ISLEC, I spearheaded the development of the first institutional intellectual property policy. So I think, Dr. Atunga, we do have intellectual property policy now. And we can look at some of the things that, that we put in there because we need to, and you put it very nicely, that the reason why we are falling below the 100% is because we are not patenting our innovations. So we are not even being known that we are actually innovating. And again, these are some of the issues that I think we need to take up with the government and also with our institutions and support people who think that some of the products that they have invented can be commercialized, and hence we protect them. The chair?
Yes, as a chair of the board of KIPRE, I'm proud to have you present such a eloquent and good product to our scientific conference. I know that there aren't many questions because you have presented this before, and there are quite a number of people here who have heard what it is. Now, I just have two questions for you. The first one is, how do you compare the behavior of HIV in baboons to that of human being? Because the products may be very effective in one species and not the other. But more important to me is, how do you get feedback on the product? Because when you go to the operating theater where smooth gel is mostly used, you don't find it as readily available as you find the KY jelly. And the reason I have myself had a comment was that it was not as thick as whatever. So I was interested to know how you get back your feedback, because this makes a lot of difference, and it may also influence how sellable and how widespread the product becomes, because the product seems to be fairly good in that it has got bactericidal and other properties which actually prevent transmission of diseases. Thank you.
Thank you, Chair. First of all, how do we compare the results in baboons and human? A product of this nature, for it to find its way into the usage by human, because there is no such a product in the market. This is actually the only African developed product of this nature. But we do have other microbicidal candidates that are in other parts of the world, which are different stages of development. We have to do human clinical trials. But what the baboon and non-human primates research does, it gives you an indication of what you should expect from human. There may be a bit of some difference in terms of dosages and all that. But clearly, if you're able to do comprehensive work in terms, especially on safety and efficacy, in baboons or in non-human primates, then it becomes easier for you to know how the situation will be in human. I know that people do not talk about the side effects that are there with the COVID vaccines. And we've had a lot of side effects with the vaccines that were given to us. The reason is that they were not tested for safety. COVID vaccines were all developed and thrown into the humans. So it is important that we get an indication of what the situation would be in a relevant animal model to human, and that is why the non-human primates research is important. However, it will not be 100%, but it will give you an indication of what you expect. Now, feedback of the products and availability. When we developed, commercialized these products after the approval, I did two things. One, I attended the Kenya Obstetrics and Gynecological Society conference on 2 consecutive years to present this data. Now, Kenya Obstetrics and Gynecological Society conference is a congregation of all the gynecologists in Kenya. And those are the people who are using these products. I'm sure even you may use it in one way or the other, but the biggest users are OBGYs. And I engaged all of them. And in fact, for us to come up with the 2 products, that was a recommendation by the gynaes, because initially we were looking at Uniplon alone, because all these 3 products are interconnected. It is the gynaes who told me that you are sitting on 2 good products. Even as you work on the HIV one, you have a good lubricant and you have a good ultrasonic gel. So that was the first thing that I did. The second thing that I did was to go to all the hospitals in Kenya. And I booked the sessions for continuing medical education. Every hospital, they have continuing medical education where all the doctors are available. So I would book a session there and I sponsor the teas and I would go and engage them. And that's how I got the feedback about the product. Viscosity of the product. The interesting about pharmaceutical industry is the competition that is there. And there are so many farmers that sponsor activities to ensure that some products are not within reach. And I'm sure as a doctor you know this. And they come up with all sorts of arguments. The point is this. We have actually compared KY jelly and Smooth-Gel, and we even published a manuscript. And Johnson & Johnson never raised an issue about it. We looked at all those issues, and that paper is available. It's one of the publications that is available. So the moment you go into the pharma industry, you expect these games. So I'm not surprised about that. But for me, the country, we have the policy of buy Kenya, build Kenya. If you have developed a product in your country, like this one, and we have a policy of buy Kenya and build Kenya, if the country does not use that product, who is going to use the product? So the challenge now comes back to us. And that's why we have to ask ourselves, how is our policy in terms of trade? As a scientist, we develop these products. We have given them for them to be tested for their safety and efficacy. They are being used. KEMSA today is actually buying more Smooth-Gel than KY jelly. And eventually, I can assure you that Johnson & Johnson will approach us, because we have now gone also to Malawi. We have introduced these products to Malawi. We've introduced to Zambia. We've introduced to Lesotho. So eventually they will have a discussion. But I would never worry about somebody who claims that is more viscous or is that viscous. One group that we've been dealing with are the sex workers. People call them prostitutes. We don't call them prostitutes. We call them sex workers because they're doing a job just like any other person. And there is a time we donated these products to them. And they were happy with them. Then later on, they approached us and they said they wanted to start selling among themselves, and we did. But after some time, they said that it has precipitates. And I had not understood what that meant. The game is very fierce. The competition is fierce, and one has to know that there are so many obstacles, there are so many challenges in the pharma industry that you always encounter. But I would want, if anybody has an issue in terms of the quality, that we can listen to. Viscosity is very easy to adjust because you just need to play around with the composition of the APIs, the components that make the product. I hope I have answered you, Chair. Yes. And the doctors who, when it's not available, sometimes, yes, you can have a lapse. Because one of the challenges we have with this country, again, is that if I import a complete product from India or from Europe, it ends up being cheaper than making it here. Because the government taxes the active ingredients, the active expenses for the products, but it doesn't, when you bring a fully manufactured product, they don't levy charges on it. So actually it becomes sometimes easier for me to manufacture in India than Kenya. That's, again, the irony about it. And when you're doing that, then sometimes you can have a lapse in terms of the shipment. We actually have time. Yes.
Thank you, Dr. Mudhara, for the very insightful presentation. My name is Dr. Michelle Imani, and I was curious, what is the effect of Unipron on the vaginal mucosa as well as the normal flora, given that it's a microbicide, especially over long-term use?
I can answer that question through a publication that we made. We actually did a very thorough analysis of the effect on the normal microflora, and we have a spectrum of all the microflora that we looked at, and also the mucosal region. And the effect on the mucosal region was actually done by a surgeon from the US who was visiting. I'm forgetting his name, Dr. Mwenda. I remember his name. He's the one who actually did that analysis, a gynae. But we do have a publication that shows that it doesn't affect any of the normal microflora and also the mucosal region. It was called Mustafa. Yes, Dr. Mustafa, yes. Any other questions? The more questions you get, the better the products get. Asanteni sana. Thank you very much.
Let's appreciate Dr. Mwidera once again. So we want to go to the next presentation. The next presenter is Professor Jerome Kim, who is coming to us from International Vaccine Institute. And this morning is going to give a presentation on the vaccine landscape and the role of IVI in facilitating manufacturing capabilities in Africa. Professor Jerome Kim is an international expert on the development and evaluation of vaccines and is the Director General of the International Vaccine Institute. He has an unparalleled experience and success in translating innovation to impact, working with the key national and international stakeholders, creating a common understanding of the value proposition, developing and executing creative strategies for sustainable growth. At IVI, he led and grew a team whose mission is to discover, develop, and deliver safe, effective, and affordable vaccines for global health, from a small Korea-based international organization to one with a global footprint and diverse and expanding funding. IVI's overall cholera vaccine is used around the world to prevent this deadly diarrhea disease. IVI's typhoid conjugate vaccine, tech transferred to SK Bioscience and PT Biopharma, both have received national approval. The SK vaccine has been pre-qualified by WHO. Prior to joining IVI, he was the program manager for the US Army's Advanced Development Program for HIV vaccines and led the only HIV vaccine trial to show protection against infection, RV144, the identification of correlates from the trial, and the analysis of viral sieve effects. Professor Kim was also the principal deputy of the US military HIV research program and the chief laboratory of molecular virology and pathogenesis, Walter Reed Army Institute of Research. Dr. Kim is a distinguished visiting professor, Seoul National University. He is also an adjunct professor at the Uniformed Services University and the Graduate School of Public Health, Yonsei University, and is an honorary professor, University of Rwanda and the University of Hong Kong. Dr. Kim graduated from the University of Hawaii with high honors in history and highest honors in biology, winning the Arthur Lehman Dean Prize in Humanities and the Library Prize in Pacific Islands Area Research. Dr. Kim received his MD from the Yale University School of Medicine and completed internal medicine residence and infectious diseases fellowship at Duke University Medical Center. He has authored over 350 publications. I only have 20, so I need to work 5 times more to get to 350, and has received numerous awards, including the John Mayer Award for Research Excellence, US UHS 2013, Department of the Army Research and Development Achievement Award for Technical Excellence 2013, and the Medal of Honor for Civic Merit, Republic of Korea. So let us appreciate him and welcome him to the podium. Professor, welcome.
The problem with being the shortest person to present. Good morning, and thank you for the opportunity to speak today about the work that IVI has been doing to support vaccine manufacturing, particularly sustainable vaccine manufacturing in Africa. Oops. The slides were up, but here we go. Briefly about IVI. IVI is an international organization of which Kenya is a member state. Our mission is to discover, develop, and deliver safe, effective, and affordable vaccines, to which a fourth element has been added, empowering vaccine equity, impact, and sustainability globally. What you see in the bottom part of the panel under discovery, development, delivery, and epidemiology and surveillance are the things that the 290 employees of IVI do on a day-to-day basis. And I think one of the most important things is that each of these components is trainable. So we can teach along the entire value chain, from the laboratory to implementation to policy. And at the bottom, you see the diseases that we work on. The ones under discovery are in the laboratory, the ones under development are in human clinical trials, the ones under delivery have often been approved, but we are trying to either generate real-world evidence or trying to generate new and appropriate, better uses for the vaccines. And under epidemiology and surveillance, and this list is not up to date, is the ever-changing number of pathogens that IVI is looking at, because when you're working on unincentivized vaccines, and it could be a vaccine against, for instance, leptospirosis, we don't really understand the true burden of a disease which is global in its distribution, which does kill people, but whose symptoms and manifestations are often nonspecific. These are the flags of the member states. There are 60 in total, and the World Health Organization that have either entered IVI or are in the process of joining IVI. This is where we work around the world, and in a sense, IVI is almost one of the best kept, unfortunately, secrets of global health. You can see that we work in Latin America, Sub-Saharan Africa, South Asia, Southeast Asia. The headquarters is in Seoul, and until 2022, that's where we were. But in 2022, we opened the regional office in Sweden, a country office in Vienna. Last year, we opened the regional office in Kigali in Rwanda, and we're hoping to soon open the country office here, which is meant to be a major center for managing projects based in Africa. And we intend to use that center, we call it AVEC Africa, Advancing Vaccine End-to-End Capabilities in Africa, to train people to do the work of vaccine development from end to end, which is the way our project managers run vaccine research and development. You can see as well the Hong Kong Jockey Club Global Health Institute. It's a very long name, but it really represents a new type of investment. The Hong Kong Jockey Club, as funny as the name sounds, is an organization that makes money on betting on horses initially, and then subsequently on betting on football or soccer, as we say in America. It is the second largest charity in Asia. Why? Because all of the money that's gained from those two sports goes into the charitable purpose of the foundation, which until this grant had been used exclusively for work in China and Hong Kong. This is the first grant that allows overseas components, the first grant with overhead, the first grant on a research and development project. And we're hoping that the Jockey Club becomes much more active in advancing research and development around global health, particularly in this time of disruption in global health funding. This is a complicated slide, but these are largely bacterial pathogens that IVI is working on in various stages of discovery, development, delivery, or epidemiology and surveillance. And the reason I put the slide up is where you see an IVI symbol, we are working on the value chain. And you can really see that it stretches from end to end. And it's this end-to-end focus that is really critical. It's one thing to be a brilliant professor with a vaccine, a potential vaccine, in a university laboratory. It's quite another to take that out of the laboratory into manufacturing, into clinical trials, to work with regulators and regional immunization technical advisory groups to get that vaccine made, approved, used, and then to work with ministries of health and finance to implement vaccination programs. So we work across the value chain. These are the bacterial vaccines. These are the viral vaccines. There are also some platforms, and actually on the bottom of the previous slide, you'd have seen MAP, microarray patches. So we are working with several companies that have these patches. One of the patches is in human clinical trials. Actually, we'll see the immunogenicity data next month, which is really exciting. Why? Because these patches are almost self-administered. You no longer require a nurse to administer the vaccine. They can be applied and removed after 5 minutes. The initial concept vaccine is hepatitis B. It was what we had available that was already pre-qualified. But we know that it works with lyophilized mRNA vaccines that are stable at room temperature. That means a lot, because it means that a patch, which has a very small volume, can be put into a supply chain that has less requirement for cold chain than others. It doesn't necessarily require large campaigns. These can be distributed more easily and more readily by other organizations. And this is the kind of innovation that will help us get to the vaccination targets that we need and may help to overcome some of the resistance to the needle and syringe vaccinations that we use currently. IVI has made vaccines. So like a small biotech company, we have developed an oral cholera vaccine, a typhoid conjugate vaccine, and a non-typhoidal salmonella vaccine. These are vaccines that started in the IVI laboratory and have been moved through pre-qualification with funding typically from the Gates Foundation. But I want you to look at the cost. This is the end-to-end cost to the Gates Foundation. It wasn't 500 million, it wasn't 1 billion, it was 28 million. So you can actually develop vaccines for less than the 500 million or $1 billion number that is often thrown around. Why? Because we have to be efficient about the way we develop the products, the way we test the products, the regulatory strategies that we use in order to get the products approved. And then the manufacturers. Shankal was Shanta, which is an Indian biotechnology company taken over by Sanofi. Sanofi subsequently, maybe 2 years ago, gave up the vaccine. Actually, one of the nice things is Sanofi actually transferred back the technology to us so that we could update the process. But in the same time now, GCBC has taken on that, and it is now pre-qualified again. So it's possible to develop a vaccine for cholera. 200 million doses of the vaccine made by Biologics are the cholera vaccine in the stockpile. So when you hear of a cholera outbreak, or if there's a cholera outbreak in Kenya that is requiring cholera vaccine, it's likely that you have a product that was developed initially at IVI. We also have initial funding for what we call vaccine number 4 and vaccine number 5, and actually now vaccine number 6. Vaccine number 4 will probably be a vaccine against Klebsiella pneumoniae. We are working on it. We're in the process of purifying the polysaccharides and planning the conjugations, and if there is some interest, these are, again, things that can be taught or tech transferred. So IVI does, and you saw the addition of empowerment to the slide, and you can see that the International Vaccinology course, which is a simple 1-week course in how we develop vaccines, has been attended by over 5,000 people. In the last 3 years, it's been provided in 3 different sites in Korea, Stockholm, and Kigali simultaneously. And the time zones are complicated, but basically the morning lectures from Korea are taped and played in the afternoon in Stockholm and Kigali, and the afternoon lectures that are given sometimes in Stockholm or Kigali are played in Korea as well. So we are able to use the 3 sites in order to educate people in the 1-week course. I think even more important now, since Africa has 46 1-week courses in vaccine development, is the work that we do with the Ministry of Health and Welfare of Korea and the World Health Organization. This is GTHB, Global Training Hub Biomanufacturing. It's meant to be the first stop in a process that leads to qualification to do mRNA vaccine manufacturing. So the first of the 2 courses is a 2-week course, an advanced vaccine development course. The cost of the course and the room and board are entirely paid for by the Korean government. The Korean government asks that the countries that sponsor or put students forward, and if the students are approved, pay the cost of travel. Over 1,000 individuals now have gone through the 2-week and 3-week course. The 3-week course is a more complicated course. Actually, there's a course going on now covering the GXPs, good clinical laboratory and manufacturing practices, which are the basis for the quality that we require in order to have vaccines approved for global health. There are as well hands-on courses. So Korea is very lucky in that they've developed a capability to do wet training in actual biomanufacturing in facilities designed for the purpose of for teaching manufacturing. And there's actually a course that's planned for the end of this year. There are 2 or 3 courses. They're a lot more expensive and complicated than the 2-week and 3-week didactic courses. Actually, I should point out too that if you go to gthb.net, you will be able to download all the course material because we've made all of it available online. The second part is the work that we do in global health security. This is the GHA Immunization Action Package, and then below, ASEAN has a vaccine security and self-reliance program, and we've been working extensively with the government of Thailand and the ASEAN countries on AVSSR. The final part is around technology transfer, and IVI was founded with a belief in technology transfer, that vaccine technology should not be limited to just high-income countries or just the large vaccine manufacturers. So we have in fact done 14 technology transfers of 6 different vaccines, developing country manufacturers around the world. The DCVMN, the Developing Country Vaccine Manufacturers Network, is actually having its annual general meeting over the same period as this meeting, unfortunately, in Bali. So many of the IVI staff are attending that meeting. But I think this is the best illustration of what we can do. So we have partnered very extensively with the Korean government. Our headquarters is there. The Korean government is the largest state funder of IVI. And we have worked with them. So in 2010, Korea was informed by the United States that the vaccine that they had purchased from American vaccine manufacturers for pandemic influenza could be confiscated by the American government and used for national security purposes in the United States, which should sound familiar because this is exactly what happened during COVID. But Korea took that lesson and started to think, and IVI worked with the Korean government to develop something called Vaccine 3.0, which had as a basis being 80% self-sufficient in vaccines and the number 5 vaccine producer by 2030. So remember, in 2010, Korea was 20% self-sufficient and the number 11 vaccine manufacturer. But it's not like you can wish to do vaccine manufacturing, you have to plan it. So Vaccine 3.0 created a vaccine research center, created 2 pilot manufacturing plants, one for bacterial, one for viral vaccines. The bacterial vaccine plant is now also mRNA vaccines. In 2018, working with the Gates Foundation and MMV, we worked with the Korean government to create the Right Foundation. The Right Foundation is 50% Korean government, 25% Gates, 25% Korean companies, and it funds, it provides funding to get things out of laboratories into clinical trials, up to phase 3. In fact, it has pre-qualified products now. So the small investment of the Korean government, which is 50% of the Right Fund, has leveraged over 3 times more additional funding from other sources. So as a way to get innovation and commercialization of products going, things like the Right Fund are very important. We'll come back to that. The next step was to, if you're going to have a vaccine industry, you have to have people who can work in it, and you can't just make copy vaccines. You want to be on the cutting edge of vaccine development. So you have to fund these research and development hubs that will train the next generation of PhDs and will train technicians to work in a biotechnology industry. And that's what these 3 things, Vital Korea, KMVAC, and then the GVL, which are really large, multi-year, hundreds of millions of dollars over 5 to 7 years, to train people to develop new technologies that will now be able to work in a successful biotechnology industry. And then by 2021, in the middle of the pandemic, the K-Vaccine Hub. And K-Vaccine is a $2 billion investment in vaccine manufacturing. But it importantly contains the Global Training Hub in Biomanufacturing. So working with the Korean government and their commitment to disseminating the ability to manufacture vaccines, we were able to move them forward. And Korea is now 50% self-sufficient in vaccines, with a target of being 80% by 2030. So it's possible, it's a multi-year process. It involves not only technology and building factories, but importantly, the human resource development necessary in order to have innovative, sustainable manufacturing. So this is IVI in Africa. As I mentioned, the IVI regional office opened at the end of 2024. Professor Nkechi Dembi is the regional director, the deputy director general for the Africa regional office. And you can see it's based in Kigali. It has a number of different strategic engagements. It's very importantly trying to develop a capacity to do strengthen human capital development and human resource development around sustainable manufacturing. And you'll see how that applies later. It's been a part of several grant applications, and you'll hear about some of them later. The AVIC Africa office was intended to be here in Kenya, and we're simply waiting for the host country agreement to really get it started. The Board of Trustees of IVI has made an investment in Africa, which is very rare. Usually we try to get funding from other people, but these are internal resources that are being used to set up the regional office and the AVIC Africa office. And Professor Faith Osier, right there, is going to be the new head of that office when it opens, hopefully next year. So I'm going to go through this very quickly because it's just to make the point that many people have not heard about IVI and the work that we do with multiple organizations in Africa, but there are a lot of projects that we're doing, and we're really more focused on implementation, on execution of projects. And maybe that's been a problem, but you can see there are a lot of IVI projects across laboratory work, clinical trials, surveillance, effectiveness, policy, advocacy, more in policy, advocacy, and effectiveness and surveillance. And again, there are a lot of projects. And the goal is to use the Kenya Project Office as the place where these projects are run, so that we can train people in all the things that we do in order to get a vaccine from the laboratory into a patient's arm. These are capacity building projects, and again, as we've ramped up work through the Global Training Hub, work on ARQUA, which is a Lassa fever phase 3 clinical trials site preparation, funded by CEPI. I mean, all of these things are creating a cadre of people who can work in a biomanufacturing industry or in academia in Africa. So, towards a more systemic approach enabling sustainable manufacturing. So I think everyone knows that the target that Africa CDC set is to be 60% self-sufficient in vaccines by 2040 from a target of less than 1% in 2019. And there are reasons to do this. COVID taught us many things. And one of the things was that the ability to manufacture vaccines, the ability to translate innovation into things that make a difference, should not be restricted to 1 or 2 regions of the world. So, strengthening pandemic preparedness, importantly, tackling endemic diseases, because one of the big questions everyone raises around vaccine manufacturing in Africa is, what are they going to produce that isn't already made somewhere else? Well, we can tell you there are 4 million people who die every year from diseases for which vaccines could be made. HIV is one of them. TB is another good example. Malaria, better malaria vaccines, but also endemic diseases of high burden that exist in Africa for which no vaccines exist are not going to necessarily be manufactured by an Indian company, certainly not by a European or American company. And who's going to make those? Well, that's a decision that governments here, in conjunction with research that's developed by African investigators, should make. But this is the problem. So after the pandemic, nearly $5 billion was committed to Africa for vaccine manufacturing. And you can see there, that's just the list of actual, I think I can use that, no, can't use the laser point, under vaccine manufacturing and tech transfers, $2.3 billion. It's a little unevenly distributed because the things that are really important on the other side, field surveillance, laboratory research and discovery, vaccine development and clinical trials, vaccine registration, are all negligible compared to vaccine manufacturing. How can you have a factory without people to work in it, without all the upstream elements that go into informing what the choices are for what vaccines to make or what biological products that we need to make? And how are we going to fix that? So we came up with this Advancing Vaccine End-to-End Capabilities in Africa, or AVIC Africa initiative, that would work with partners in Africa across the value chain, because that's what IVI does, and to use what we can do to train others here in order to be able to develop products, to have the managers and technicians, the project leaders, the people who can lead research and development efforts. Because, we know, and, the U.S. military HIV research program has been working in Kenya, at Kemri Kericho, and Doug Schaefer is here, I think I saw him in the back, since the late 1990s. There are people who can do clinical trials, who can do laboratory work. There are people who can lead efforts. And the question is there a way to empower them to become leaders? So this is the framework for action developed by what used to be PAVM but is now FOM. AVIC Africa actually reinforces 4 of the 8 parts of the framework, and so this would have to be a collaboration with the African Union and Africa CDC. Taking this as an example, IVI is doing a tech transfer of oral cholera vaccine, simplified oral cholera vaccine, to BioVac in South Africa. If this is successful, this will be the first successful technology transfer of manufacturing of a drug substance in Africa since yellow fever in the 1930s. We are now at phase 3. We've assembled the funding with BioVac to do the phase 3 study, and we've worked with them across the value chain. This is really an important first step, but the funding is really complicated. Wellcome Trust, Elma Foundation, Gates Foundation, and now the FCDO. And it just gets to the idea that one of the reasons that we can partner effectively with organizations here is that we understand the nature of this multilateral funding that is necessary in order for us to move forward. This is just a roadmap, again, thinking from end to end. AVIC Africa wants to train people to do what IVI does, but located here. So bringing people in, and we always talk about retaining good personnel. The whole point of this program is to train people so that even if we lose them, they're going to be going somewhere else and contributing to important work to develop the ecosystem to support sustainable manufacturing. So people always say, well, but how does this relate to all the other programs? You know, are you sure you're not doing something that other people aren't doing? And in fact, there are a lot of other people doing a lot of the other work across the vaccine value chain. Africa CDC and IVI work across the entire value chain. Others work in individual parts of it. We think that a systematic approach, taking vaccine products and developing those products from end to end, the thinking around that end-to-end development is what is critical. And that's what AVIC Africa plans to do. But we hit 2025 and a bump. And I think everyone knows the first half of 2025 was a miserable time. I mean, disruption, not only from the US withdrawing from WHO and Gavi and other things, but also countries that have been traditional funders of global health programs reducing their contributions because of national security pressures or others. And in fact, if you start to think about all of these, what are we really faced with now? We're faced with global health going from a unipolar, largely US and maybe Europe focused, to a much more multipolar. And I think that we always discount what Asia is contributing, but their economies and their commitment are relatively sustained. But again, the idea here is that going from global north to global means that we have to collaborate, we have to cooperate, we have to think about sustainability, because depending on a single source of funding, we know can be dangerous. And we've heard African leaders saying this as well, which is really important, because leadership is the first part of this. The second, it's one thing to lead global health from Geneva, and it's been very effective. I mean, we've made tremendous progress over the last 2.5 decades in reducing childhood mortality. But should the leadership in global health be moving to the front lines, lead from the front? And I think that that's starting to happen. And again, we're hearing this from African leaders who are talking about the need for regional manufacturing, the need for a cholera champion, the need for sustainable health investment in Africa. We're hearing these things. So leadership is one of the things that people are starting to talk about, and the next step is ownership. Because as USAID was withdrawing from PEPFAR, for instance, countries were stepping up and saying, you know, we should have funded a part of this ourselves. And some of the countries, like Nigeria or South Africa, are moving forward to do exactly that. So ownership doesn't necessarily mean that you have to pay for all of it. But there should be some commitment from the country so that there's a sense that the country has a piece of this overall stake. And most countries do. It may not be obvious, but many of the in-kind contributions that countries provide are often ignored. And I think that this is going to be important. I think the last point here is one that IVI is very committed to: closure of the global commons. So we talk about global health products as being something that it should be available to all. But we also hear that, well, if we develop new products, we're going to keep them to ourselves. I think that what we want to do, and what IVI would encourage, is that, you know, ideas and innovation generate IP, but there has to be some balanced access to that intellectual property that is shared among low and middle income countries, or even with high income countries that happen to have problems. But we have to think about these things now rather than during an emergency, because if we are in a crisis like we were during COVID, we know what's going to happen. Those ideas, and then we've also are aware that data generated in Africa does not necessarily stay in Africa or analyzed by African scientists or research institutes, and then samples are taken out and analyzed outside of the continent. And should that always be the case? We're hoping that the first idea is to try to take advantage of the thinking that's going on and to put funding around it. So we'd like to propose, or we actually have proposed, something called Achieve Africa 2.0, Accelerating Health Innovation and Excellence for Vaccines and Biologics Development in Africa. And this was actually something USAID asked us to put together. So between the US Military HIV Research Program sites and IVI, we put together a $100 million proposal that went into USAID on the 28th of January, just as it was closing. And our colleagues and collaborators said, this is such a great, it was supposed to be African-led HIV vaccine research and development. So African-led. And they said, you know, this construct was so important. Is there a way that we can get $100 million to fund it? And we thought about it. And we thought about it pretty hard. And we thought, well, maybe it can't only be focused on HIV anymore. It may have to include other things. But what is really important is the ecosystem for sustainable biomanufacturing, innovation manufacturing, which is more than manufacturing itself. It's everything, including the training of the scientists and technicians who will work in these efforts. European and American partners are important, but they should be consultants to the work that is led by Africa. So again, we were targeting $100 million, which should sound familiar. And we were talking to Professor Drew Weissman. Drew is the person who got one of the 2 Nobel Prizes for mRNA. And he said, well, why don't we put $25 million down and make an initial contribution and see if African countries will take a stake. He made the announcement at the 75th Regional Conference for Afro. So this was August in Lusaka. And he didn't say 25. He went and he announced that they were contributing $50 million, and Penn would donate the IP to low and middle income countries. So ownership of the IP and the initial funding. But there needs to be some other components. So the idea was to have 5 African countries contribute $1 million a year for 5 years each to be the stakeholders in this. Rwanda, Nigeria, and Zambia have committed. We're working with Kenya, with Ethiopia and Egypt. Those are the ones that are closest to actually being additional countries that commit to the $5 million over 5 years pledge. And that will be core funders of this effort. So there will be African ownership of this effort. And then once we have the $75 million, the plan was to go ahead and approach the European Union, and not IVI, or not the US military HIV research program, but the African leaders who pledged the funding. So President Hichilema, President Kagame, Minister Pate from Nigeria would lead this effort to go to the European Union and say, we have $75 million. It would be great if Europe could match this, because you know, Europe pledged $2 billion. They actually haven't used all of it. Okay, my time is up. So this is Penn's contribution, the African contribution of $25 million, unknown contributions from others, but in fact, Rwanda, Nigeria, and Zambia have committed. We have another commitment of $18 million, so we're actually above $80 million in committed funding without yet going to the Gates Foundation or the European Commission. We really need, what we're waiting for are 2 additional countries to step up and commit. The second idea is something called the Bright Fund, which is similar to the Right Fund that I mentioned previously, that $71 million from Korea, Gates, and Korean partners has leveraged $200 million in additional funding. It's actually gotten products going, and we think that we need a Right Fund for low and middle income countries. And we're calling this Bridging Research Investment in Global Health Technology. And in fact, the Minister of Health from Indonesia has decided that he will commit to be the champion. Discussions are pretty advanced with India and Brazil to be contributors to this fund. And originally we were targeting $200 million. Actually now the amount has increased to maybe $300 million as a result of the discussions that we've had with the core funding partners. Could Kenya be a part of this? Absolutely. Would this help? Would this help to get the product from Kemri, a diagnostic for instance, commercialized and give Kenyan companies the ability to manufacture things that they wouldn't otherwise be able to do? You know, it's one thing to be in the US or in Korea where you have venture capital or stock markets to raise money. What the Right Foundation and what the GHID in Japan have done is they can work with global health products. They can work with novel diagnostics, new drugs to treat malaria or TB, and move them forward into phase 1, phase 2, and phase 3, and really kind of incentivize the development of products that are specific for the needs in low and middle income countries. The third idea is something called BridgeVax, and I won't go through that since I think I'm out of time, but thank you very much. This is just a summary of the things that IVI does.
So thank you very much, Professor Kim. I think it was a very serious presentation, and I saw a lot of figures in $1,000,000, and I was thinking that this is a very huge investment, and we need to take it very, very seriously. So in the interest of time, I think we can allow 2 questions. Then we can take them, and then we'll break.
Thank you.
Thank you, Professor Kim, for such a wonderful talk. My name is Kennedy Okeo. I work at Purdue University and Jackson Laboratory in the US. I've been working with colleagues here to strengthen capacity building in basic research, which I think is a very critical link toward achieving the vaccine development vision that you told us today. And I believe this is something that we should continue doing. So my question is, under the vision that you outlined, do you have any practical steps towards supporting research infrastructure within the country, which I believe needs a lot of strengthening before even we talk about basic research? And if you have those practical steps, could you outline them, please? Because I believe this is critical.
Yeah, that's a great question. And it really gets to the investment that countries make in research and development, which is not as high as it should be. And so one of the things about Achieve is that, and we can't fund all aspects, but the commitment of University of Pennsylvania's funding is not only for vaccines, but for therapeutics. So if there are therapeutic mRNA products, like they're making tremendous advances in vivo gene sequencing, gene editing, which means that you might be able to correct certain genetic problems. Could we work on that? And so Penn has agreed to allow people to come to Pennsylvania to be trained and to retain the intellectual property when they go back. The advantage of therapeutics is that it also then keeps the capacity to do mRNA manufacturing warm, because you make a batch of vaccine and then nothing happens until the next pandemic, people aren't going to be ready. But if we have therapeutic products that are being made, developed, tested, then we can. I think the other aspect of this is we're thinking about the entire research ecosystem. So for instance, one of the initial proposals was for KIPRI to be the Center for Primate Research, animal research. And so, we're using this as an example, we're funded with Afrigen to develop a Rift Valley fever vaccine. Could that vaccine, not GMP because Afrigen doesn't have that ability yet, but a non-GMP product be tested at KIPRI? And we use that funding to reinforce the ability of KIPRI to do that. For small animal work, it'll be at Noguchi in Ghana. And again, the idea is to create these centers where, if there are products to be tested, if there are products that come out of African laboratories, they will go into the animal testing facilities. Samples will be shipped to the designated clinical research assay development centers, again, in Africa. Data will be sent to a single data site at the University of Rwanda, which is going to be augmented with a biostatistical training program and bioinformatics and AI in collaboration with the University of Bordeaux, which is committed to do this as well. So the idea is to build capacity along this lane, but also keep as much as possible within Africa itself. It's not perfect because, again, it's only $100 million. It may be more than that, but it's still not going to be enough. And that's why we thought that the contribution from the countries was really important. I mean, you think about $5 million out of a total of 100 million, it's a 5% stake, but it is a stake. And it puts people, again, this is African-led. The political leadership is the funding. The scientific leadership, the leadership of the effort will be based in Africa. I mean, the data and samples will be here. We know that Penn is committed for the IP to be here. So all of the elements that we've been talking about, that people have been worried about, are now going to be accounted for. But could you put more funding into this or other efforts, national efforts or regional efforts to strengthen research? Yes, absolutely. But there has to be a start. And we have to be able to show that it will be successful. And the $18 million is actually funding for human resource development. Again, the whole key to this is having people who can do this. So, sorry. And
thank you very much, Professor Kim. for sharing the lessons from IBI. You have partly answered my first question, actually, which was to do with the main driver for success. Because during the COVID period, COVID pandemic, there was a lot of, political statements, and I think looking at some of the pledges, I think putting the money into the vaccine manufacturing, I think has been a challenge. There are a lot of countries interested, but when you look at the actual investment, it hasn't really matched that, those political statements. So I wondered, what do you think is the main driver, particularly to maintain that interest? And specifically, which is also my second comment, is the target set by the Africa CDC that increase the vaccine manufacturing from 1% 2019 to just about 60% by 2040. We're already in 2025. And I'm just wondering whether we are on track or off track in terms of the target set by Africa CDC.
Yeah, so I think that's so I'll take that question first because it's the most difficult one. It's hard to see when we're in 2025. The progress we hope will be more than just linear. That as there's the development of more and more capability, human resource capability, that the ability of, say, Kenya Biovacs, or the new effort in Ghana, or the ShieldVacs in Ethiopia, to move forward will be augmented. The question is, how many of these centers of manufacturing do you need, and which ones will survive? And can some of them be both therapeutic, so if they're mRNA, like the mRNA manufacturing hubs that the WHO has set up, can they not only be vaccine, but can they be therapeutic as well? And I think that's where the commitment to train people with the IP reverting back to low and middle income countries becomes so important, because there's ownership of the IP, which is a really critical piece that was missing before. And so, we're in the process. We would like to get the first organizational meeting of the stakeholder countries together to work on the governance and other really key questions, like what are the first things we want to work on? Getting that together requires 2 more countries, and maybe it's artificial, maybe it's fine if we only have 1 more country, but I think the commitment of the countries is a part of the political leadership and ownership that's so important. Scientifically, we know that there are scientists in Africa who can lead these kinds of efforts, and we need to put them in place, but we are waiting, and we have the funding from Penn and others, but we really want to have the commitment so that it looks, so that this effort is Pan-African and really has the commitment from governments. So.
Thank you, Professor Kim, for that very insightful and very serious presentation, giving a roadmap on how we can manufacture vaccines and so forth. So now we are almost going to tea break, and before we do that, I would like to introduce a few people, and I request them to come in front. They are our partners, starting from Pauline. Pauline, kindly come here. And the other team, which was with Joy, come online here, 1 minute to introduce yourself. And then after that, we shall go for tea break. When we come back, the room will be partitioned into 2. This side will be A, and the other side will be B, and then you can join as part of the program. So, I will start with Pauline, then they will say their names 1 minute, then we break.
Thank you, Doctor. Good morning, ladies and gentlemen, all protocols observed. I'm Pauline from Safety and Security, and on behalf of the hotel management, a very warm welcome to Emara Ole Sereni, Nairobi. Where we are, the meeting room is called Mara Ballroom, and in event of an emergency, we all shall use the fire escape doors at the back, which are clearly marked from outside. The fire escape routes are clearly marked to our fire assembly point located outside the hotel main parking. Today, there is no planned fire drill, so we'll be expecting 0 interruptions during our session. For medical attention, the hotel has a clinic that operates from 8 to 5, and from 5 onwards, emergency doctor on call. Washrooms are outside the 3 elevators. You walk straight to your right, or behind this wall. That's where we have gents, ladies, and people of determination. We have a smoke designated zone that is outside the terrace facing the Nairobi National Park. Your lunch will be served at level 3. You will be issued with 2 meal vouchers, green one and brown one. For brown one, you'll go to level 3. The restaurant is called Acacia, and for the green one, we'll go to level 10. The lounge is called Acacia. While attending this meeting, please make sure you put on your badge. It's very, very important. Make sure at all times you display your badge, and also make a point of knowing the neighbor seated next to you. For access control and identification, it's very important for security to know the participants attending this event. We are going to partition the hall, so as we sure you carry all your belongings. Also, I would also want to emphasize on another thing. Do not leave your items unattended. While attending this conference, make sure you be with your items all the time. Security are here, dedicated, just to make sure for our safety and security is guaranteed. Once again, thank you for choosing Emara Ole Sereni. Enjoy your tea break.
Good morning. My name is Orina Jeremiah. I work with Africa Biosystems. Yeah, my name is, it's okay. Great. So my name is Jeremiah Orina. I work with Africa Biosystems. We are part of the sponsors of the AIBBC conference. Africa Biosystems is the channel partner for Thermo Fisher Scientific. And we support the entire range of products from Thermo Fisher Scientific, the Bioscience Division from Sambo Prep, thermocyclers, proteomics, cell culture, the real-time PCRs, and Absolute Q digital PCR, and all the master mix with real-time PCR. Then we have Sanger sequencing solutions. Additionally, we have the sequencing solution. That's the Ion Gene Studio S5 and Genexus. Then we also have the chromatography and mass spec division. Our stand is outside there, so I'm welcoming you all to come and have a conversation in case you have any need of these solutions. So we support from products, service and maintenance for the instruments, and we also offer technical support. So once again, we are thankful as Africa Biosystems for an opportunity to be part of supporting your research work and even to supporting this conference. Thank you.
Good morning, everyone. My name is Irene Moriuki, an application specialist at Exela Biotechnologies Limited, and I'm glad to be here. So Exela Biotechnologies is a local distributor for Beckman Coulter. Beckman Coulter is one of the sponsors for this conference. So we do provide solutions for life sciences products for research and also clinical setups. We do provide solutions for flow cytometry. So we've been supporting the just concluded flow cytometry workshop at MKU. We offer flow cytometers for both use in clinical and also research institutions. Other than that, we do support centrifugation under Beckman Coulter. We do automated liquid handlers. We also do support particle counters. Other than Beckman Coulter, we do work with other different manufacturers. So we are offering oncology solutions for molecular and also NGS and real-time PCR kits for precision medicine and targeted therapies. We also do support laboratory consumables, so we do offer a wide range of laboratory consumables. We also do offer laminar flows, biosafety cabinets, incubators, shakers, a wide range of laboratory equipments. And so we have both outside, and would request you to kindly pass by and get more information about us. And we're really happy to be part of this conference and for having us. Thank you so much.
Morning, everyone. My name is Maureen Ongonge, and I'm an application specialist from Change Biotech. Change Biotech is a company that offers end-to-end solutions. It's a one-stop shop where you'll get your solution from sample collection to sequencing. And to enable this, we have different partners whom we partner with to bring these solutions to Africa. We have QIAGEN, which is our amplification and DNA and RNA extraction partners. We have QIAGEN kits, we have amplification products. These range from first generation PCR to third generation PCR, which we'll showcase outside there on the digital PCR. We also have sequencing solution, and we are accredited to partner with Oxford BIOTECHNOLOGY, which offers short reads to long read sequencing, and this is able to be accessed anywhere. You can carry the portable sequences from the lab to the field, and you can enjoy sequencing and get any read that you want. We have these solutions outside there in our booth. You can visit us and enjoy and talk to us and learn more about our solutions. Thank you so much for allowing us to come and showcase what we have. Thank you.
Good morning. I stand between you and tea. My name is Cecilia Rumberia from F&S Scientific, and we are also part of the sponsors for this workshop, and we're really proud to be a part of the team that are joining you here today. And I'm very glad for the conversations that are happening here today. And here now we are going from the lab to the translation side, yeah? And at F&S Scientific, I'll just reiterate what the other partners have said. We also support end-to-end. We have solutions that allow you to do your research more efficiently. We have a full suite of team, from application specialists to the engineers who will maintain your systems. So we also offer capacity development. We don't only support you with instrumentations and reagents. So we are glad to be partnering with a lot of you who I see here, who we work with. And we do have a booth outside, so do stop by and hear what we offer. So we are the local support for Illumina Technologies, who are, I think most of you will know Illumina when I talk about that. And not just Illumina, we carefully select very high quality partners who will enable your research here in Africa. So thank you so much to the organizers for allowing us to even speak to you and to be part of the conference this year. Thank you.
Thank you very much. So while we are taking coffee or tea or lunchtime, please let us visit the booths. They have some nice products. I've been seeing them, so that we can also, because they've come to showcase, just to hear what they have, new technologies, I think is very important. Secondly, we have 2 serving points for coffee. So we'll have one serving point out here, the other one the other side. So let us not overcrowd in one place. And finally, I want to introduce Dr. Mercy. Kindly stand up, Dr. Mercy. For the posters for day 1, Dr. Mercy, raise up your hand. People are struggling to see where Dr. Mercy is. Others are looking at the other side. So Dr. Mercy is with us. So make sure those ones who are displaying their posters today kindly see Dr. Mercy so that the posters are ready. We shall only break for 15 minutes. So by 40 minutes past, we should be back, so that the session chairs now will take control of the sessions. Thank you, and I wish you well during the coffee time.
Two, 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 two. 1, 2. Sound check 1, 2. 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 1, 2. 1, 2. Sound check 1, 2. 1, 2. Sound check 1, 2, 2, 1, 2. 1, 2. 1, 2. 1, 2. 2. 1, 2. Sound check 1, 2. 1, 2. 2, 1, 2, 2, 1, 2. Hey, 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 hey. 1, 2, 2, 1, 2. 2, 2, 1, 2. 2, 1. Check 1, 2. Check 1, 2. Check 1, 2. 2. Check 1, 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 2. 1, 2. Check. Testing 1, 2.
Check. 1, 2. 1, 2. Check.
pointer huwezi tumia. Huwezi tumia pointer juu. Panel huwezi point panel na light.
So siwezi point kitu sasa.
Ni ngumu.
So naweza kaa na kitabu hapa hivi.
No problem. So utasimama tu hapo. But your control. Hii uko able kuona presentation yako yote. Utatu point huko. So hii ndio forward, hii ni back.
Hii ni kuzima?
Hii ni pointer, lakini sasa unaona ina dissolvingi ndani. Hii ni panel, unlike ile projector you can. But hii ni panel. Panel huwezi. So utaangalia
We would like to start this session, so I'd like people to be seated kindly. This is room A, and B is next door, okay? Okay, so we are congregating now. Okay, so we are in session 1, room A, and we're looking at immunology of parasitic infections and progress in vaccine development. Today, I'm excited. We have a wide range of speakers. And this morning, I want to introduce Professor Faith Osier. Faith is a long-term friend. We actually did our PhD together. She's reminding me we used to dance together, and when we were in the earlier days. Anyway, Professor Osier is the chair of Immunology and Vaccinology, Faculty of Natural Sciences at Imperial College in London. She's also the co-director of the School of Convergence for Health and Technology. She's currently also the director of Chanjo. Hopefully you'll get to hear more about that in due course. Her research team's vision is to make malaria history through vaccination and also focusing on vaccine candidate discovery. She's won multiple awards, such as the Rosati, Pfizer, the Sofia Kovalskaja, and the UKRI African Research Leader. She's passionate about emerging African scientists as key agents of change, delivering health innovations in our continent, and we urgently need those. So with that, let me allow Faith to come and give her presentation, and Karibu.
So good morning, everyone, and thanks to the organizers for inviting me. It's an honor to be here. Hopefully my slides will miraculously appear, and we can get going. Yeah, so thank you. So I'm a Kenyan scientist. I'm based in London at the moment, but I'm really passionate about capacity for science and for the young people to build their skills and have impact on the continent. So today I'm going to tell you what we do, what we've been doing for malaria for quite a number of years, and it involves human challenge studies, which I'll describe in a minute. Next slide. My pointer isn't moving. Yeah, so just a few points about malaria. This audience doesn't need much. We have over 200 million clinical cases every year. The numbers actually haven't changed much for almost 10 years. We've got 600,000 deaths every year. Malaria can give you lifelong disability. It has a huge economic impact. And a lot of the interventions we use involve drugs. And so drug resistance is a problem. Next slide. We do now have 2 malaria vaccines, and you'll have heard about them in the press. Kenya has been one of the implementing countries. The first one was RTSS, and the next one was R21. And so whilst this is really exciting, there have been some problems that the vaccine is good, but not as good as we want it to be. So in this example from a BBC communication, if you vaccinate 10 people, only 4 of those would be protected, and that was with RTSS. And that's gone up to 7 with R21, but there's still challenges, and that's why we continue to work on malaria. Next slide. And so the issues are efficacy. So you remember with the COVID vaccines, which have been mentioned before, efficacy was going up to 90%, 99%. For malaria, we're not yet there. There's questions of durability. Once you get the vaccine, how long are you protected for? On average, this is about 6 months. So what? Who cares? If you give it for 6 months, it means you need to get a shot every 6 months. If you're a mother in a rural situation, you're a busy parent, you have to take your child to the clinic every 6 months. This is logistically challenging for national immunization programs, logistically challenging for families. So we want to develop vaccines that give protection for a long period of time. There are issues of strain specificity. So if you think about COVID, we started with the alpha virus, then we went to beta virus, then we went to Omicron and sub-Omicron, et cetera, et cetera. The parasites, the viruses, they change. And so when you design your malaria vaccine, it might work well for the first strain, but it doesn't continue to work well for the other strains. And so those are some of the challenges. We had problems with supplies. That's why RTSS was rolled out only in a few places. It's increasing now. Things are getting better with R21. And that's why the talk of vaccine manufacturing is really important, because that's when you can control your supplies. And people are also dealing with monoclonals now as another way to prevent malaria. So what my group has been doing has been working in Kilifi in Kenya, where we understand that humans can acquire immunity. And I won't go into too much detail because of time, but the basic point that this graph is showing is that young children are at high risk of severe malaria and they can die from it, but their moms and dads and aunties and uncles in the same house also get the infection, but they don't get sick, they don't die. And that's because they've acquired immunity. And so what our group has been trying to understand is what are the molecular markers of that immunity? What is it that the moms and dads have learned that they haven't passed on to their children that could protect them from malaria? And so in the 60s, people did this experiment where they took antibody from people who had become immune to malaria, and they passed it on to people in hospital who already had malaria parasites. What you see on the graph, I don't think the pointer works, but what you see on the graph is the parasitemia going down. So what you could imagine is that you've come to hospital and you say, hey, my child is sick with malaria. And I tell you that instead of giving you artemisinin drug, like we're using in Kenya at the moment, I give you an infusion of antibodies from people who've had malaria many times. So that's exactly what they did, and they had a great result. They showed that parasitemia went down, and actually clinical symptoms also resolved. And that lesson from there is that antibodies are really important components of immunity to malaria. And so the life cycle, you can all look up in your textbooks. I won't go through it, except to say that the work I'll show you is on the blood stage, which is in a circle there. The blood stage is when you get the chills and rigors and you feel that, whoa, 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 I've got malaria. When you're bitten by a mosquito and it goes to your liver, there are no symptoms. Everything's okay. But when it gets into your bloodstream, that's when you have the clinical symptoms of malaria that are dangerous. And so if you look at this, just try and go back. Can you play that video, please? It might not play, but in any case, this is a video of your bloodstream, and there's merozoites. There we go. So the merozoite is the malaria parasite. The red cells you can see, it finds a red cell, it attaches to it, it orients itself, invades the red cell. Once it's inside, you'll see it multiplying. Lots of daughters are born, and these come out and they infect other red cells, and that's how you get the cycle of fever in malaria. And so when you look at that cartoon, this is the merozoite. This is the stage that invades the red cell. What we've been trying to understand is, where do the antibodies bind? We know that the antibodies are protective, so where do they bind? And it has a surface coat and organelles at the apical prominence that secrete antigens, which are all targets, which can all be targets of protection. So in our group, we've been trying to understand what could better vaccine candidates be, especially in the blood stage where you have the clinical symptoms. What are the correlates of protection? What could I measure in you that would tell me that you're not going to get malaria the next time you're infected? And how exactly does it work? What are the mechanisms? And so I'm going to tell you about a human challenge study. In a human challenge study, you actually volunteer. You say, Faith, I want to join your study. When you come along, we give you an injection of live parasites, sporozoites. We've done these studies in Kilifi in Kenya. I'm sure you've heard about them, but if you haven't, here they are. Once you get your infection, we know that they go to the liver. They stay there for a few days. After 6 days, they emerge into the bloodstream, and we monitor that infection by qPCR. When we did this study, the participants were resident with us at Pwani University in Kilifi, so that we could take care of any medical emergency should it arise, and we also had all the ethical clearance, all the consultation, etc. And so we could respond well to the study. And so the results were fantastic. First, I'm going to skip through this. This was how we recruited our participants. If you've never had malaria and we measure your antibodies, we'll find nothing like what you see in green. If you've had malaria many, many times, like you live in the bundus, in shags, as we call it, what we found from Kilifi is people are packed full of malaria antibodies. So what we did is we recruited participants from Western Kenya and from coastal Kenya, where we know the distribution of malaria. So we had people who had very low levels of malaria antibodies. These are all Kenyan people, but no malaria antibodies. We had people with very high levels of malaria antibodies, and we had people in between. And we wanted to understand, does having a range of antibodies protect you against malaria? These studies are always done in healthy young adults, etc., and we chose people with a range of previous exposure to malaria. So what you'll see on this graph is the most fantastic result. If I infect you now with malaria parasites and you have no immunity, your malaria parasites will grow exponentially. And that's what we observed in a proportion of the study participants. But the magic is here. In other participants who we treated in exactly the same way, they gobbled up their parasites and we could not detect them at all. So these people were immune. You give them the infection and the immune system deals with it. We had another group in whom the parasites appeared on day 6 as expected, but they didn't grow exponentially. They cleared that infection. So again, these people are also immune. They're seeing the infection and telling it, ha ha ha, goodbye, I can deal with you. And then we had a final group that were also interesting, that the parasites did appear, but they sat on them. You know the way you can have papers on your desk that you know you're supposed to do something and you don't do it, right? So these guys just kept the malaria under control. It did not reach the threshold where we needed to treat it. They had no clinical symptoms. So in the first group, we didn't let the parasites grow forever. As soon as the participants said, we're sick, we treated them, and they went home. And in the rest of the groups, these participants did not become sick. They stayed till the end of the study, and then we treated them at the end, and then they went home. So now I show you the immunology. So this is published. If you want the details, it's almost 4 or 5 years old now. So now we begin the immunology. We measured antibodies to the merozoite. This is the blood stage parasite that I showed you entering the red cell. What you can see, the last 2 columns, PCR positive and PCR negative, those are people who controlled or cleared their infection. What we see is that they had higher antibody levels than the febrile and non-febrile group. Those are people whose parasitemia grew and grew fast. So the next thing we did is we said, what kind of antibody is it? Is it a IgG1, 2, 3, or 4? And the answer was that it was IgG1 and IgG3. Now, those are called cytophilic antibodies. Those antibodies communicate with other cell types in the body, and I'll show you how. And that becomes quite important for understanding the mechanism. So we set up all these assays with PhD students, who I'll show you here. These are all Kenyan PhD students who are working with me in Germany. The first assay is a blocking assay. We call it growth inhibition. And it looks at the ability of antibodies to block entry. And that's the gold standard for malaria. But we had known that that wasn't really a strong correlate of protection. So we looked at all these FC-dependent assays, which look beyond blocking. They look beyond the engagement. They look at the engagement of the FC part. You know, your antibody looks like a fork. It's got this part of the fork that identifies your pathogen. And then it's got the long arm of the fork that engages with other cells of the immune system. So the first one is complement, monocytes, neutrophils, monocytes again, and natural killer cells. So what these PhD students did was they developed all those assays, and then we had a look at the data. And the data was fantastic, and you can look at these papers that are referenced here. But what we found was that the blocking assay did not discriminate people who were able to control the infection versus those who were not. However, the FC assays, if you look at all these graphs in detail, they're very significant. That was a correlate of protection. If you look at the same data in a different way, in this forest plot, the hazard ratio, when it's 1.0, it means there's no difference between groups. But what you can see is our point estimates are really high, between 0 and 0.2. So we had a very strong effect with tight confidence intervals. All the functional assays are now on the y-axis, and the blocking assay is at the top. That was also interesting, but it wasn't significant in this analysis. And so when you measure an assay in the lab, you measure one thing at a time. We added those things up because in your body, all the cells are there at the same time. When you add that, we're calling it the breadth of the response and how it correlates with protection. People who had function in up to 4 assays, you can see them at the red line on the top, they were the best protected. So the more that your antibodies were able to recruit these other members of the immune system, the more protection you had, and that correlated very nicely with the amount of antibody. So this is breadth at the bottom, and that's the amount of antibody. High levels of antibody, high breadth of function. And so what I've told you is, I've shown you a different way of working using a human challenge platform. Actually, I've been doing this work for almost 30 years. This is the best data I've ever got, because for the first time, we were working in the actual model of malaria and in very controlled circumstances. So that was actually transformational. We found our correlates of protection. It was simple. It was antibody binding. We found that that antibody is most effective when it's engaging with other cell types in the body. We found that that was a good correlate, while the one we've been working with all along, which works well for viruses, that wasn't our best predictor of protection, and that we could now identify people with clinical immunity. So how does this then relate to vaccine design? So what we had done is, the malaria parasite has a lot of proteins, so we went through a process of prioritizing the proteins to find out which ones are doing the business, which ones are the important ones. Again, I show this because these are all Kenyan scientists. These people have all moved on to do other things in life, but they were at various stages at the time they did this. We produced this chip here in Kenya, and we used it to study our responses in the human challenge study. Because of time, we've prioritized a number of antigens. This was our study statistician at the time. He's now working in Roche here in Kenya. But we prioritized antigens, looking at different methods of analysis. And then we said, with the antigens that we have prioritized, can we demonstrate function? And so this work, again, was done in Germany by Misha. These are all the functional mechanisms, and again, now we're looking at a specific antigen, merozoite surface protein 1. We're seeing very strong correlation with protection, and that's published as well. You can look it up. And so what next? And here's where we go into the translational parts of the work. We applied for more funding to look at the quality of antibodies, because what the data was showing us was that, yes, you're focusing on the antigen a lot, but if you don't get the right kind of antibody, then your vaccine doesn't work as well. So now in the group, we are focusing on glycomics, understanding the sugars that surround the antibodies, because those relate a lot to the cytophilic function. We're studying the B cells, and we're also looking at the epitopes of the antibodies. But in addition, in relation to vaccine manufacturing, we've also gotten funding to do a small-scale project where now we're taking the candidate from the work that we've done into manufacturing, and this is in line with what Jerome Kim from this morning was saying, that we want to build African capacity. We believe that there's a pathway for African scientists to develop products, do their research, take them through a development pathway so that they're designed in Africa, they're developed in Africa, and that we can go through the entire pathway, including business, entrepreneurship, and innovation. These are all themes that are strong in this conference, and I hope that this demonstrates that it's actually possible to do. So this is what we're doing in Kenya. We're upgrading small facilities. We're not building big factories. Upgrading a small facility. This is a process for mRNA vaccine design into which the malaria vaccine candidate that I showed you is going. And we hope that in the end, we'll be able to do another human challenge trial in Kenya, this time vaccinate the participants and then challenge them and see whether actually all the science is making sense. And so with that, I'd like to thank the people who take part in the challenge. To do the challenge study takes a village, so this was all a while ago. We started this kind of work in 2014, so it's it's been a long time. The faces will have changed, and this is the research group that generated the data, and thanks to the funders for funding the work. Thank you very much.
Thank you very much, Faith. It's good to see that malaria vaccine development is moving into the continent and using controlled human studies. So thank you for sharing your research work. I'm just going to allow time for maybe brief 2 questions. If we can have a microphone. Do you have another one? That's okay. You'll test it. Brief.
Thank you, Prof, for such wonderful presentation. Based on, you are talking about developing malaria vaccine, but can you comment about malaria species? Can it affect, because we have, I think, 5 species. Is it going to affect malaria development? Because you have only talking about strains.
Thanks for the question. That's why the young people in this audience are here. This work is Plasmodium falciparum. And you're correct, there are 4 other species. This is specific to falciparum. So there's more work to be done, and that's why we need the younger people to take on and continue. Thank you.
Thank you very much, Professor, for your wonderful presentation. I just want you to throw more light. I was thinking when you said that there is a problem of the parasite, your vaccine usually have a short life and doesn't work for a very long time. I was thinking that the ability to characterize different proteins and really study them very well, will it not be possible to look at the particular protein that gives the capacity for that mutation or whatever structure that enables that parasite to mutate and sort of that capacity? I don't know whether you're thinking in that line.
Yes, and actually people have been thinking in this line for 100 years. This is a continuous problem for viruses, also for parasites. So yes, there's more to be done. You know, one can only go as far as one can go. But yeah, with new technologies, I think this should be possible. But this is an active thing. People are now trying to use AI to try and see how can we beat the parasite at its own game. But the parasite has been in existence for hundreds of thousands of years, you know? So yes, people are doing it. We've also done it, you know. This is as far as we've got, but it's a continuous problem. Thanks. Yeah.
Thank you very much, Faith, for your presentation. I hope you'll be available for, there are a few more questions that were from the audience. So Faith, you'll be here to take them at some point? That's a question. You'll be here around, right? She'll come back. Okay, fine. So she'll be there. So let me now invite our next speaker. Okay, so our next speaker is Dr. Bernard Konoy from the Marquette University. He's a biomedical scientist and also specializes in immunology of infectious diseases, particularly malaria and emerging pathogens. He holds a PhD in life sciences from Ehime University in Japan, and he's also done some postdoctoral work. His research utilizes genomics, bioinformatics, and advanced immunology assays to explore naturally acquired and vaccine-induced immunity. He also leads multidisciplinary teams where he's focusing on training immunity and immune imprinting in endemic populations, aiming to enhance vaccine and immunotherapy development for malaria and other global health threats. He has over 75 publications. And lastly, he's passionate about building capacity, and through that, he's actually serves as a member of the Federation for African Immunology Societies and representing East Africa. So over to you, Dr. Konoy, and thank you.
Thank you, Lucy, for the introduction, and I hope I can see my slides. And I'm happy. Thank you for the chance to present what we are doing at Mount Kenya University. I'll highlight briefly some of the studies we are conducting, and this is an ongoing work. So we still believe there is so much that's still not to be done and so much we can collaborate on. So my topic we'll be touching on malaria during pregnancy, specifically the immunology aspect of the same. So Professor Faith talked about malaria in general, and that's a very nice introduction. So I can build on that and talk about malaria in pregnancy. This is when pregnant mothers get infected. There is something that happens in the placenta. The parasites, they are able to hide what we call sequester in the placental tissues. And by doing that, they are absent in the peripheral blood, meaning that when mothers go to the clinic and they do the normal malaria test, they don't have the parasites. So the doctors think the mothers are okay. The mothers feel they are okay because the tests were negative. But when they deliver their babies, they realize that they have these complications. For example, very tiny babies, what we call low birth weight, or miscarriages, stillbirth, because the parasite in the placenta is affecting the normal function of the placenta. So this burden, we see that, allow me to pull this one out so that I can keep turning as I present. So this burden is highest in Africa, and the outcome is more than 10,000 deaths per year, stillbirths, and more than 500,000 low birth weights. This is a substantial burden, considering when mothers get this type of problems, it don't just affect the mothers, it affects the whole family. It affects the husband, the older children, and the burden becomes amplified in the community. Okay, so what do we know about this? We know that the problem occurs when these parasites are able to hide in the placenta. The infected red blood cells express unique proteins called VAR2CSA, and these proteins are able to attach to other proteins on the placenta that are basically and mainly expressed in the placenta, and this makes them to hide there. And the outcome is inflammation in the placenta, and this affects how the placenta functions. Also, we see other innate immune activation and also production of cellular disruption. What is the outcome? Maternal anemia, maternal death, abortions, preterm births, stillbirths, low birth weights, and these effects go beyond the first few days of life. They can have lifelong effect on the children. What are the challenges with this case? Right now, there are no tests that can detect placental infection. So as I said earlier, when the mothers come to the clinic, they test negative, but actually they are infected because we cannot get the placenta and test. We cannot pick a piece of the placenta because the gold standard of testing for placental infection is looking at the placental histology. And we do not actually know what is happening at the interface between, the placenta belongs to the baby, and what is happening in that placenta? How is it affecting the baby's development? What are the immune responses happening in that phase? And we don't have vaccines to protect against malaria during pregnancy. So these compounds and makes it complicated that although we know there's a problem, we are not able to tackle it. So what are we doing at Mount Kenya University? So we have been doing these studies in the western part of the country to try to really have a systematic way of dissecting the whole issue about placental malaria. We conduct cohorts in Bungoma, in Webuye, and also we also conduct studies in Thika to help us really to see, is there any difference between malaria endemic regions and malaria non-endemic regions? We collect placentas, we collect maternal blood, we collect cord blood representing the baby, and the outcome is to try to develop tools that can help us detect placental infection or understand which proteins, apart from the main one called VAR2CSA, could be critical to prevent against infection. At the same time, we want to understand what are the immunological changes or what does the dysfunction of the placenta affect the fetus that is developing. So, over the last several years, we have been trying to see which is the best way to test against placental malaria. So, Kobe and the team, they have tried to use different approaches, trying to use PCR, histology, RDTs, using PCR targeting the placental tissues, placental blood, peripheral blood, and there's a lot of discrepancy. We don't have one unique approach apart from histology that can tell us that this mother is infected. And histology can talk about, it can be negative, or it can be current infection, meaning that when the mother was delivering the baby, she was infected, or it can have chronic infection, meaning that the mother has been infected for several months within the 9 months of pregnancy, or it can be past infection. Maybe she was infected in the first trimester or second trimester. And this has an implication on how the baby develops. So, there's a test that is done for pregnancy, the HCG test, where we use some urine, and using that, you can tell that somebody is pregnant. And can we use such an approach where we are looking at, are there some products that are coming from the placenta or that are being induced by the infected placenta to inform us that this mother is infected? So we partnered with our collaborators at Uppsala University to try to understand this. We did high throughput analysis by comparing serum samples collected from mothers that have placental malaria and those without placental malaria. And the idea here was to try to pick even tiny amounts of differences that could be existing between these malaria-positive mothers and negative mothers, so that we can be able to deduce or have an idea that something is happening within the placenta. So what we have found is, at least we were able to pick several proteins, inflammatory proteins, that could be indicators of the inflammation in the placenta.
and be indicative of placental infection. And one of the positive aspects of it is that these proteins or these inflammatory molecules seems to be increased or present at different trimester, either first trimester, second trimester, in the positive and the negative mothers. So we are keen to follow up with this. Now with our growing cohort of well-characterized samples from these malaria-endemic regions, so that we can further pinpoint which of them and how we are able to develop tests based on the ECG understanding. But we can test, but we should be able to do more. I think the best is to protect against infection. Do we have a vaccine that can target and protect the mothers? We don't have. We have one called VAR2-CSA that carries under development, but we think there may be other molecules that can be targeted. So we are using two different approaches here to try to identify new molecules that can be targeted as vaccine targets. So we are using the immunoscreening approach that Faith well described in her presentation, and we are also using reverse vaccinology, where you use bioinformatics AI tools to be able to down-select target several antigens, which we can now do functional studies and subsequently characterize them as vaccines. But one of the biggest challenges with malaria proteins is most of the malaria genes are AT-rich, which makes it very difficult to make recombinant proteins to help you do this characterization. So at MKU, we have this approach that we use to make recombinant proteins of any kind. We use what we call the wheat germ cell-free system. This is where I use the germ of the wheat, which has all the requirements of making proteins for the plant. But now we become parasitic. We are tapping into that and using those resources to make proteins that now we can use for further characterization and be able to understand the different proteins and how they could be involved in protective immunity. So basically, you have you make your plasmids of interest with genes of interest and express them by mixing them with the wheat germ. You go home in the morning, you can have idea proteins when they are ready. So with this, we recently did some studies to look at the immune profile of pregnant mothers, where we targeted about 700 different proteins that represent those expressed in the placenta and also in the merozoite stage, as we saw in the previous presentation. And here we have every dot represents a protein, and there are different types of proteins, the blood stage protein, merozoite stage proteins, and those that are expressed on the surface of the infected red blood cell. And the idea was here is to have an unbiased approach. We don't know which is important, but if we make it very unbiased, we may be able to pick some of them that could be targeted for vaccine. And what we observed is that mothers that have had several children have an upregulation, or they have abundance of antibodies targeting 2-3 major proteins that we think are important. So one of them is a RON4 protein that is involved in the infection of the red blood cells, and others are those expressed on the surface of the infected red blood cell. So we normally have several proteins that are on the surface. They are called the surfins, rifins, stephas, PFE-MP1s, and they are highly polymorphic proteins, but they are key for sequestration. Even in cerebral malaria, they are the main proteins that are involved in those different aspects. So one of the other things we used the bioinformatics approach and using literature and focused on this protein called CSA ligand. CSA ligand is a protein that co-expresses with VAR2-CSA, the main molecule involved in the sequestration of the parasite in the placenta. And previous studies have observed that CSA ligand co-expresses, co-localizes in the same region with the VAR2-CSA. And we wanted to know, can we target this? Is it a good target? Is it a good molecule? Can we develop vaccine candidates based on it? And what we did is to use our cohort to see what are the immune responses in different trimesters of the mothers when they are infected. And we could see a gradual increase of antibodies as the pregnancy develops in the first, second, and third trimester. But one of the key questions that has always been a major problem with malaria vaccines is the issue of polymorphisms. The proteins change, or the parasite has a way of changing the proteins, if I could use those words. So here we have a comparison between VAR2-CSA at the bottom right. This is a leading target for vaccine against pregnancy malaria. So the peaks you see, they represent how variable that protein is. On top of that, we have ADSL. This is a housekeeping gene, which is supposed to be highly conserved because they are essential for the survival of the parasite. And on the left, we have, at the bottom, we have AMA1, which represents one of the vaccine targets, and we have CSA ligand. And from this, if you compare the peaks, you can see that CSA ligand is as conserved as ADSL, one of the housekeeping genes. And we have actually identified the target region that interacts with VAR2-CSA, and we would like to further work on these as potential targets. If the placenta is infected, how does it affect what goes to the mother? If you remember in Faith's presentation, she gave us that figure showing us that people acquire immunity when they are pregnant. They acquire immunity over time. But we know that in the first 6 months of life, children rarely get infection because they receive antibodies from the mothers. But now, with an uninfected placenta that is disrupted, has vascular compromise, local inflammation, and also has some level of dysfunction, is it still able to let the antibodies pass from the mother to the baby? Are the babies still protected? So we have done some preliminary work to see whether the passive transfer of these antibodies is happening as should be happening. And what we can see here is that mothers that are infected seem to have a lower rate of transfer of antibodies. It means that when you compare between positive mothers and negative mothers, we see lower rate of transfer of these antibodies, suggesting that infection of the placenta could be affecting how the antibodies are transferred. This is just for malaria. Does it affect other vaccines? Does it affect other diseases? Something we are looking at, and this should be in our next presentation, in our next meeting. So just to conclude my presentation, what is the way forward? Where are we going next? We are keen to see how this exposure of the parasite affects how the babies respond to childhood immunity. We want to see how does it affect how their neurocognitive effects are affected, and also how does the coming in of different parasites, as our previous, somebody asked in the previous presentation, like vivax, nocci, affect the placental status of infection. So I'm very happy to show you a picture of the guys who do this work. It's a big team at Mount Kenya University. We work on different aspects, and for those of us who came in for the workshop, you saw where we do this work from, and we are happy to collaborate. We open our doors. We believe that alone we cannot do much, but together we can do a lot. So thank you so much. Our fathers are there who have given us money to do this work. Thank you so much.
Placental malaria? Because you've talked about the CSA, but what about the HA, the hyaluronic acid? There might be multiple receptors involved in adhesion. So I don't know if you looked at that at all and your thoughts around it.
That's a very good question. So far, we have only focused on CSA, but we are also exploring how to use glycoproteins as indicators of placental infection. So we haven't looked at that molecule, but something that we can look at, but we are looking at the glycoproteins in totality as indicators of the placental infection.
Thank you. Any question from the floor?
Thank you, Dr. Kanoi and the team. You are doing a good work. I'm Dr. Kosio from Maseno University. You fully explored the parasite genes that may change with respect to semesters. Did you also consider the human genes, specifically to red cell related, like the hemoglobin S, hemoglobin C, and also maybe thalassemia and G6PD? Thank you.
Yeah, that's a very good question. I know Paul works on that space. We are not looking into that. We are looking at the specific receptors, but we have collaborators we are working with to try to identify new receptors that we don't know about, because there may be some other receptors that could be essential for the sequestration or the invasion of the red blood cell, as we saw in the video in the previous presentation.
Okay. If there are no more further questions, I'm looking around. Okay, so thank you so much, Dr. Kanoi. We appreciate your talk. So we're now going to move on to 2 online presentations. So I think as they're loading, the first one will be from Dr. Mutakai Ngara. Allow me just to read a bit about him so that you understand who the speaker is. Okay, so Mutakai Ngara is a postdoctoral fellow at Karolinska Institute, and I think he also works with the Mount Kenya team. His PhD applies novel single-cell RNA sequencing to the development of Plasmodium falciparum, the malaria-causing parasite. He's currently working on an integrated computational approaches to understand the immune response to malaria infection in field settings. Towards this end, he's currently establishing single-cell transcriptomics techniques in remote settings in Kenya to allow for experimental profiling of primary host immune responses. In collaboration with various groups at Lund Diabetes Center, he's also working on single-cell profiling of pancreas with an interest in determining gene regulation in type 2 diabetes. So we welcome Dr. Mutakai. I hope you can hear us.
Yes, I can hear you. Can you hear me?
Okay, yes, we can hear you. Please go ahead. Thank you.
And the slide, you can see the first slide?
Yes, we are there now.
Just to confirm that we can move.
Yes,
that's moving?
It's moving, yes.
Okay, great.
Thank you.
All right, thank you, Dr. Lucy and the team for this opportunity to present some of the work. Of course, it would have been better to be at location, but due to commitment, it's been really difficult to travel. But I definitely know you're having a great learning experience and discussion. So for this work, I'll just briefly share what we're trying to do with the single-cell technologies. And our aim here is to understand the host response towards malaria infections in remote settings. The idea here is to apply this technology within location. So as probably some of you may be aware, is that most single-cell technologies are usually applied off-site. So you collect your samples, then you ship them somewhere, and then you do the FACS sorting, or somewhere where you have, a collection of sophisticated tools like, say, FACS sorting or a droplet system. But there are also techniques that are coming up that I will highlight, and we're already starting to see some of the data. So this is ongoing work, but we are starting to see the data, and it's quite convincing that we can actually apply these technologies within the remote setups. So just to give a background, we know that host response during malaria infection is a complicated process. It's driven by the parasite factors, the geographical location, and host factors. And this may give you distinct phenotypes of infection. So you may get submicroscopic infection, and those are quite important. You can get microscopic infection, and then there can also be variation in terms of severity. So you may have severe infection, or some people get moderate and mild infections. So the fundamental question that's been ongoing for several decades is in what drives this different displays of the infection. So people living within the same location, exposed to the parasite in the, at equal levels, what drives their distinct phenotypes? Or even within the same person, you may get instances when they have severe infection or mild infection. What goes on in their immune systems? Because we believe immunity is fundamental to this process. And this was already proven in 2002. So towards this goal, we want to develop the technology to make single cell work within location, so in these endemic settings. But then after we show that the technology works, we want to take it to the next phase where we start to understand the biology. So what immune cells correlate with this distinct phenotype of malaria infections? Do we have, say, some TCR markers, so T cell receptors that are definitive of the phenotype we see? So phenotype here is a case of what kind of malaria, how severe the infection is, for instance, what parasitemia levels do we see in the host, for example. And then what clonal dynamics do we see? So what kind of T cells or B cells do we see? How do they change over a course of infection? So in the long term, that's the goal we want to achieve, but now we are starting to already prove that the technology works. So that was the key thing, and this is what I'll mostly share for today. So towards this goal, we established a longitudinal cohort that we followed for a whole year. This was based in the Homa Bay County. This is closer to the lake, where we established a longitudinal cohort of around 300 individuals. And this is across age range, so from 5 years to 65 years. And then we followed them for 12 months. Every month we get a sample. So if we get a venous sample this month, and then the following month we get a capillary sample. So it's in that alternating fashion. But every 2 weeks, we had our community health workers who went to the community and talked to our cohort members, interviewed them, surveilled them, their malaria, their health status. What was, did they travel, for instance, to a different location, for instance, so that to make sure that any infection that occurs is within this location as well. As you can see from the plot, the bar plot, we balance for things like sex. So we have equal number of males and females. The age ranges are different. We have recruited more slightly younger age groups, just to increase our chances of getting clinical episodes of infection. So, but just to emphasize that during this period, we follow them closely. We collect a lot of malaria-related data. And this is quite key when you're designing some of these studies, because the metadata also will help you make sense out of it using computational approaches, for instance, or statistics as well. So just to give a feeling for the cohort, this has run successfully. And what we see is that we had a high retention of cohort participants. So we have close to every month or every 2 weeks when we were doing the interviews or sampling, most people showed up. And this I can attribute to the many years of working this location that was started back with Professor Kira and Professor Jesse from Mount Kenya University. And so we piggybacked on this relationship that was established over a year. And this was quite helpful when we were doing these cohort studies and to ensure that it succeeds. As you can see, we do, during the testing, we use 3 approaches. So the RDT, this is rapid diagnostic test for malaria, microscopy, and PCR profiling. And the PCR here is species specific, so we know whether it's P. falciparum, it's a P. ovale, or any malaria. So or it's a mixed infection as well. What do we see now? Across the cohort members, we see high incidence rates in younger children, which is not surprising, but it's always good to see that your data aligns with what's expected. We also would see seasonal variation. So we know that there are 2 peak seasons of transmission in this location. And when you look at the data over time, we see that during May period, there's a high peak of infection, then also in around October because of the rain season. But then there was microscopic infection, but the frequency is low. But significantly is that we have a lot of submicroscopic infections. And this is quite significant in this era as people are trying to go towards eliminating malaria. Then submicroscopic infections are quite important because these are the ones that sustain the transmission. These are individuals that are infected, they don't go to the hospital, they are healthy, and if it's an adult, it's okay. For most of the time, they are fine, but you can imagine the more vulnerable groups, younger children or pregnant women, they're more exposed to this. So That's why it's important to address and actually to understand what goes on in submicroscopy infections as well. Majority of the infections were Plasmodium falciparum, but we had a few cases of P. falciparum, P. ovale also that occurred, but majority of them are P. falciparum driven as expected. And we have up to cases of mixed infection of up to 3 species. So if, for instance, you can see you have cases where you have P. falciparum, malariae, and ovale. But also notable is that we've never detected any case of vivax infection. This in the cohort that we've looked at. So we're doing 3 tests. We're doing RDT, we do PCR, and then we also do microscopy. And so this may allow us to phenotype what kind of phenotype of the disease someone is experiencing. So for instance, you may have submicroscopic infections that are RDT positive or RDT negative. Then you can have microscopic. Some of them may be most of them tend to be RDT positive, but some cases, we've seen cases where they are RDT negative as well. So based on this, majority of cases, of course, were negative because of, I think, better control, but then you have up to 11% that are microscopic, which is still substantial and helps us to achieve the goal we are driving towards. A lot of submicroscopic infections that I mentioned before as well. So based on this test that we've done and monitoring the trajectory that someone experiences during the 12 months follow-up, we can use a computational approach to classify groups. So when we apply an unsupervised approach to classify the cohort members, so each row here represents a cohort, and then what happens during the 12 months, and then you use computational approach to group these individuals, we come up 3 major blocks. We have individuals that are frequently infected, and they tend to have microscopic infections as well. So you can see in the lower section of the heat map. We have a substantial number that are submicroscopic, and most of these are also symptomatic. And then we have those who have moderate infection. In certain instances, they have microscopic infection. Other cases, they have moderate infections. Some may be submicroscopic or not. So these are the 3 major groups. But I think you can appreciate that there's a lot of complexity that goes on in this data set. So an individual may be microscopic at a certain point, then they become negative, and the next time they are submicroscopic. And if we assume that the biology is still constant, then we can ask our question, what's going on in the immune system? Right, and also get to know the complexity that drives this. This is just to give you a feeling of what we are seeing, for instance. So each row here represents an individual that was monitored for the 12 months, and the different instances where they have. For instance, here you see submicroscopic, they're negative, then the next time they're microscopic, RDT negative, and then the submicroscopic infection twice. And so you can see the complexity that I'm talking about. And then you also have an additional issue of species complexity. So some people are P. falciparum, others are mixed infection. And so the data is quite complex. The metadata is quite important. And also, if we integrate all this, then we believe we can sort of try to understand the biology that goes on in the whole system. So this is just so the cohort is done for the moment, and now we are moving to the next phase of applying the single cell analysis. So the first phase, we wanted to try this protocol. Can it really work? And what we adopted is what we call split-seq. So the power of split-seq is that one of the key defining things in these technologies is how you separate your cells into individual reactions. So for instance, most people may use FACS sorting to isolate the individual cells into wells and then do the reaction in the wells. Or you may have a droplet approach. So with 10x, for example, 10x chromium is quite popular for that. You have your cells isolated in a droplet, and then the reverse transcription reaction takes place there. But what's attractive about the split-seq based method is that you use the cell itself as the reaction center for the reverse transcription reaction. So what you do is you fix your cell, then you permeabilize, and then you introduce the RT reaction reagent inside the cell. So the cell itself reacts as the container for the reverse transcription. So after you do that, then during this RT reaction, you introduce these unique barcodes. So you get the reverse transcription reaction with the barcode, and then you pull your cells together and then do another round of of barcoding. And so if you do like up to 4 rounds of this, then what you end up is a unique combination of this barcode. And the idea is that using computational approaches, we can pull apart each of the transcriptome that it belongs to an individual cell, right? And the power of this, just a key thing, that's why this is useful for our settings in Kenya, for example, or remote areas, is that you don't need a fax sorting machine. You don't need a fax sorter or any microfluidic system. This is quite key for us. And I think it would be useful for to adopt this kind of technologies, especially in the remote setting. What we see now, so this is an initial data set of around 10,000 cells that we've done. And what you can appreciate is that the cDNA libraries are how they look. You can see a nice peak here. This aligns quite well with what we saw in the other protocols. And this is also the TCR. So you can do TCR profiling at the single cell level. And we're starting to see that the profile also aligns to what's expected. So as I've said, these were individual, these are 10,000 cells. We targeted 10,000 cells derived from around 4 donors that we profiled so far. So this was just initial to show that there's a proof of principle that this can work. What do we see in terms of the sequencing has also been done. So this is, proud to say, this was done at Mount Kenya University, where we have access to the ISIC NextSeq 2000 platform, which was quite useful. And what you can see is that the libraries have high friend score. Basically, there's the probability that this read call, the base calls that we got are wrong, is maybe 1 in 1,000 chance. So that's a really good library. That's what this figure shows. And then for single cell work, you usually have these rank plots. So what you want to see is this knee-shaped plot. And this idea is that you're able to differentiate between background and your real single cell transcriptome. And we see these knee-shaped plots across the libraries that we've done so far. As I mentioned, their targeted was 10,000 cells. And so far what we've yielded is about 8,000 single cells that have passed the quality control. So it's fairly good and it works in our hands. And happy to say that most of the team members who are involved in the work, some of them are at the meeting as well. So for instance, Cedric did a lot of work. He's one of the member participants in the conference. So feel free to talk to him as well if you get a chance. So the next stage was single cell you want to make sense. So when you do, what we do typically is you remap your read, you do computational analysis, and then dimensionality reduction just to ensure that you can define clusters. But before you get there, you have to make sure that you have no batch-driven effect. So for instance, we see that there's low mitochondrial fraction, there are very few cells. So individual deaths, each dot here represents a single cell transcriptome profile. What we see that most of the single cell profiles have low mitochondrial fraction genes. And the idea is that if you have high fraction of mitochondrial genes in your single cell, then you're probably sequencing a non-viable cell. But what we see is that most of our transcriptomes are very low fraction of mitochondrial mapped reads. In terms of number of detected genes, we don't see any variation. So maybe you don't see any bias towards high, cells that have more genes detected or cells that have very few genes detected. So they're fairly uniform, the number of genes detected. In terms of donor, if you see your samples grouping by where the donor who got the sample from them, then you have to be worried. But we see that across donors, we see that they are spread out nicely. And in things like time point, when we sampled, so we looked at very many factors that may drive batch effects. And this is just a sample of what I'm showing for this. We also enriched for T cells. So there's this protocol that you can assay for T cell specific profiling. And we see that in cases where we use this enrichment strategy, most of the T cells actually group with the expected T cells. I'll show that in the next subsequent slide. So the obvious question was to identify the cell types that we're able to detect. So we get monocytes, majority were T cells, so up to 70%. We get ILCs, we get macrophages, plasma cells, B cells, and dendritic cells. Of course, it's T cells and B cells were the highest. And then they classify nicely. If you can see that this principal component plot, see that this batch is mostly T cells. And then here we have your B cells, and then you have your monocytes and dendritic cells clustering. So we're able to identify the clusters nicely using computational approaches. In terms of, we just also profile in terms of the composition per donor, what cell types do we see per each donor? And we see that most of them, we have high fraction of T cells, followed by B cells, but then there's variation between ILCs and also dendritic plasma cells, sorry. So there's variation in terms of the fraction of reads a donor. In this case, this is the one where we enriched for T cells only. So you can see that nicely, there's only more T cells that we detect, almost 90%. So this is just to make sure that our protocol works. It works in our hands, the data makes sense, and the biology starts to align before you look into the biology. If we use just all cell types put together, we look at the most variable genes, and then we classify based on the disease phenotype. Do we see any grouping? No, we don't see. Most of the classification is mostly driven by cell types. So in the next steps, what we are doing now is delving more into each compartment. So we go down to T cells, and within T cells, we have subtypes, and we can start to see how does the phenotype of the disease look. But that's ongoing at the moment. Just to mention that the key markers for these cell types also light up. So this calmodulin-dependent kinase, for instance, is CD8, CD4 positive cells. You see that it's only expressed in that compartment. We have granulosin, which is cytotoxic T cell specific. We see that lights up only in the expected cytotoxic T cells. The B cell marker, DANK1, is also only in the B cells. ILC3 marker also is in ILCs. And the T regulatory T cells, as you know, regulatory T cells is quite popular now. Then we only see them light up in a subpopulation. Within each of these major T groups, we also have subtypes. So within T cells, we see the cytotoxic, memory, and also naive T cells. B cells also can be grouped memory, B cells, age-associated. So you see that we see this data that can be broken down into more granular subtypes. Of course, we have more CD8 and CD4 T cells, and this is quite useful to what we are trying to address. So this is just to give you a feeling that the protocol works. We can do single cell within the remote setting. And this, as we move forward, we are trying to do more samples. So to just improve the numbers as well, but also show that the biology will align. So biology, we are starting to just analyze further and make sense of it and align with malaria, see what goes on in malaria. But initially, just to show that the technology can work and we can adopt it in remote setups. So from key points, we identify malaria key phenotypes. We show that this protocol can work, split-seq-based method can work. And then from our cohort, I think we're just starting to understand it. So there's still room to explore the other modalities. So what goes on at the epigenetic level, what goes on with the proteins, what goes on with the metabolites. And this has also been our bioarchive. What I'm saying is that there's room to collaborate and do much more with the data. So we will improve the scale of the single cell that we profile, and we hopefully can publish this soon. So we'll look out for papers soon. I think that's what I would say. And this work was supported by many. It's a big team. We have our founders. We have big collaboration team in Mount Kenya University, Karolinska, Japan as well. And also Dr. Lucy and Mercy, they really supported that as a start when we were starting to set up this cohort work as well. So, and many of you probably in the evidence. Thank you very much for your time.
Thank you so much, Mutakai. It's exciting to see how you're using the single cell transcriptomics to actually characterize different cell populations. We always think of flow cytometry, but it's good to bring in this other angle. So I'd like to ask if there are any questions. Any questions from the floor? So as the team are thinking, can you just guide us a little bit on how you are actually, what markers you are using to type those different cells? I may have missed it in your talk.
Right.
Just a bit about the methodology in terms of typing so we can get the different cell populations, or the markers you're using to identify different single cells.
Right. So there's a probably, I don't know if you're probably familiar with this cell typist platform that was done with a group in Sangha, I think. They've come up with a cell, like a classification model for all the cell types that are immune related. And from here, you get to see each of the cell types and the marker that corresponds to them. And then using a scoring. So here we use a Wilcoxon ranking method to see where is a marker expressed or not. So there's a bit of the statistical and computational, but I relied most of the markers, I picked them from this cell typist platform, and also this UCSC browser. They have this single cell resource that you can identify a cell type and the corresponding marker. So using that, and then you identify if they were expressed in each of these transcriptome that you're looking at. I'm not sure if this answers.
Yeah, that is answered quite well. Thank you. Thank you so much.
Yeah, right.
Anyone else? Okay. So thank you so much again.
All right.
We really appreciate your time.
Thank you very much.
Presentation.
Yeah.
Thank you. Okay, so our next speaker is Dr. Brenda Apio Okech. Is she online? You have the presentation, right? It's good. Okay. So in terms of her bio, she's the director of a non-profit organization called Uganda Viral Research Institute Clinical Research Program Initiative. UVRI CRP carries out prevention research with the fishing communities around Lake Victoria and has interest in HIV, malaria, Ebola, and cancer. Brenda has been an investigator in several studies of various disease areas, including COVID, HIV, malaria, and schistosomiasis. She's passionate about effective leadership and working towards vaccine products that promote health. She's an alumna of African Science Leadership Program of the University of Pretoria and Executive Women in Leadership Program in Cape Town. She has experience as a scientist as well as a manager of research. She has a clinical trial monitoring, both site and processing auditing experience, and has worked for several sponsors including GSK and Amanet. She previously worked for both of those, and she obtained a PhD from London School of Tropical Medicine and Hygiene in malaria technology. Brenda was trained by the WHO TDR for 1 year on malaria clinical trial management at GlaxoSmithKline in Belgium. She has also worked with malaria vaccine clinical trials for Africa Malaria Network, Amanet in Tanzania, as project manager, and for Statens Serum Institute, Copenhagen. And she's based in Uganda and supports basically also the European Vaccine Initiative as a consultant. So Dr. Brenda, we welcome you. We hope you can hear us. You need to unmute. Thank you. Nice to see you. Okay, over to you.
Nice to see you, Lucy. It's a real pleasure for me to be on this panel. And just like someone said, it would have really been nice to be in the room interacting with all of you. And I'm very, very jealous of all of you in the room. It's such a pleasure to give a presentation. And a lot of what I wanted to say, I must even say I'm grateful I hadn't shared slides. I had to remove it because I see we are time barred, but also I was going to repeat many of the same things. But like Lucy has mentioned in her introduction, I'm quite passionate about vaccines. And the reason is this simple slide kindly shared that's about TB elimination. Indeed, we know that smallpox is now eliminated, and if we are to think about a world that's free of infections, then vaccines are indeed the way to go. And from this slide, you can already see that there are so many positive effects when new vaccines come into play in terms of reducing the rates of, in this case, TB, but the same can be said about any of the diseases that we want to discuss right now. So it's quite important that we keep our eyes on this tool, even when we have so many others. Vaccines will remain quite a highlight. So one of the things mentioned, UVRI, we changed names. We used to be UVRI IAVI, but now we are UVRI Clinical Research Program Initiative. And we work with fishing communities around Lake Victoria. Not only fishing communities, we also work with healthy populations. So depending on the study that we are doing, we also have access to people who are from from the mainland as well. But this is a picture of one of the outreach meetings in the fishing communities where we work. And of course, in such a place, schisto is one of the main challenges that they face. So most of our work actually is around HIV. And in fact, our previous name even had the name vaccine program. So we've now changed to diversify and speak more to the more diversified portfolio that we are moving forward with now. So at the moment for HIV, we are mainly involved in the HIV prevention trials with PrEP, and we are involved in the cabotegravir study. We are involved with the lenacapavir, so the 2-monthly injection to prevent HIV, and also the 6-monthly injection. And we are also excited because we are soon to be part of a monthly injection study still for the prevention of HIV. For this particular talk, of course, schisto and malaria are the key areas of interest. And these are pictures from our program. And if you haven't had a chance to visit, you're very welcome. We are based in Entebbe within the Virus Research Institute. So for now, for malaria, one of the studies that we are doing, actually really just concluding, is an epidemiology study of malaria infection. And there are other countries as well involved. There's Mozambique, there's Tanzania, and Gabon as well. And the main reason for this is really to assess prevalence of malaria and also the transmission dynamics. We don't have the results yet. And all these are in preparation for future vaccine trials with BioNTech that we are really all looking forward to. So we have been involved really in a 6-monthly study. And the aim, of course, was to, as I mentioned, look at prevalence of malaria incidence. And in this case, we are looking at children 5 years up to adults all the way to 60 years. Because, of course, as you know, there are already vaccines. And this BioNTech is looking beyond the children population. And you know, just like the previous speaker shared, it's about RDT, microscopy, and also PCR. But for us, we do not have the results yet. We are just concluding the study right now. So the 2 malaria vaccines that are currently rolled out are, of course, the Mosquirix and also the R21, and they are being rolled out in many different countries. So BioNTech has an investigational product that is aimed to prevent blood stage infection. And the study that I mentioned we are part of is really to prepare for the later stages of this particular work. And part of the challenges, because you would wonder if we already have malaria vaccines anyway, why are we still trying to find new ones? It's because they are not quite perfect, and there's reason, of course, to keep looking. And one of the issues I mentioned also is the 2 vaccines that are out there are really for children. And yet there are also adults that have issues with malaria. So this is one of the reasons for the study, to really understand how big of a problem is it in the adult population. And some of the challenges to malaria vaccines is, of course, the waning immunity. It would have been nice if there was sterilizing immunity for malaria, so that, you know, after 1 shot, like the measles vaccine, you are set up, you're okay and protected for life. But this is not the case. And even for people living in endemic areas, they develop some natural immunity, but they still continue to get some infections once in a while. The other is immune evasion, and I think
Recording
other
in progress.
have mentioned this a lot when it comes to pregnancy, the virgins. All that is part of the reason why vaccines in this area have not been so successful. Then, of course, parasites themselves are complex, the life cycles are complex, and sometimes even within different hosts, and that comes with its genetic variability. Of course, climate change means that even areas that were not susceptible, more and more are becoming susceptible to some of these in a time when we all want to be talking about elimination and total prevention. However, there are also new ways that people are looking at to try and overcome this. And there is work by Claire Gutu where she used in silico methods to find hypothetical proteins that could then be used for malaria
Recording
development.
stopped.
This work is now published, and she was able to find 23 proteins, and some of these she's now moved on to testing in rabbits to really see if they are protective. So some of these new technologies are really going to be useful in us getting many products for parasite vaccines. Of course, messenger RNA technology, we all saw how it was a game changer when it came for COVID. So hopefully some of these strategies will also help the scientific world make bigger progress towards vaccines against parasites. The nanoparticle delivery is another. And for malaria in particular, there is also transmission blocking vaccines that are under development. The previous speaker talked about those subclinical levels of parasites that may not actually cause disease in the person carrying them, but then that disease could be transmitted across the population. Also very interesting is vaccines that could maybe target various stages of the parasite. It starts in the liver, it goes to the blood. So could it be targeted even when it's in another host? Then very exciting is controlled human infection studies, and there are many examples, but I'm very grateful that I have some slides that I'm going to share that are very specific to schisto. And like I mentioned, schisto is of interest to us because we work in the fishing communities. However, it's a team that's next door to us within the Virus Research Institute, and this is the MRC team. And many of these slides have kindly been shared by Moses Egesa, and they are looking at controlled human infection in schistosomiasis. As you can see, just like for any other parasite, the host immune response is very key. And of course, there are also regulatory cells involved. And all these are very important to understand if indeed a successful vaccine is to be got. Then immune evasion is also not just for malaria, but it's also very much present for schisto. And these are some of the mechanisms that are involved in evading the immune system. There are many ongoing trials at the moment for schisto in many different countries, and the team in Uganda is among those that is part of such a study. And the main idea of the controlled human infection is really to accelerate the vaccine development because you're able to tell much quicker if a product is working or not. In natural infection, you may not be so sure. Has this person who has received the vaccine actually been challenged by the infection or not? And hopefully, when we have a challenge model, then we are able to actually introduce the parasite and we can tell if the participant is exposed and then be able to tell how the vaccine is working to prevent infection. So it's consenting participants, of course, have to accept to be exposed to the pathogen. But of course, with all of this, there is usually intense follow-up. And this happens for diseases where we know about treatment and we are able to treat. So this is not possible where there is no treatment for that particular parasite. So examples here are from viruses, from malaria, from bacteria, and now for the team that's at MUL or Medical Research Council, London School collaboration in Uganda, they are working with Schistosoma mansoni. There are already several publications in this vein. One thing that is very key is that the products are well tolerated. Infection was detected using a very sensitive method and of course treated very early, and the participants remained undetectable up to 1 year after treatment. So of course there are many considerations if one is to consider the challenge model, and in this case for schisto was there prior exposure, because of course this would already influence the immune response, and we know that immune evasion is one of the things that we are really looking out for. Of course, also other infectious and environmental exposures. And for us in particular at the UVRI CRP, what we've observed is when we are doing other studies for other disease areas, the presence of schisto infection modulates the immune response, and so could result in variation of even responses to vaccines. We have a study that is highlighting where presence of schisto resulted in lower responses even to the yellow fever vaccine. So these schisto vaccines are of course very much needed in endemic countries and in areas like in the picture where I showed people who live in islands, and in their day-to-day they are really very highly exposed to schisto. So here then is another example about how it goes with schisto. For natural infection, the parasite has a mixed sex carrier, but when it comes to the challenge model, it is now only one sex, and this is the reason why we are able to actually prevent infection, because all the symptoms that are usually there when it is a natural mixed infection, the liver fibrosis, the portal hypertension, the hepatosplenomegaly, all those, stunting, anemia, they are not seen when we have the single sex. And this is the single sex that's being used in the challenge model. So that's the reason behind the success of the schisto model in Uganda. And the work is ongoing, and what it is expected to show is that, a dose of this single sex is really safe and it's infectious, but it will not actually lead to disease. And also the magnitude of adverse events is of course controlled because they are really followed up closely. The number of the Sakari in 100% of the volunteers was detectable. And there are still also some areas being explored. That's the time to infection, responses, the immune responses to these particular schistoparasites. So all those are going to be very informative when it comes to looking at vaccines. In this case, schisto, but also for other parasites where the challenge model is available. So I want to really end there and very much thank the MRC team who very kindly shared their slides with me, MedBiotech, Thomas Egwang. I also really want to acknowledge BioNTech SE, who are the sponsors of the EPI study that we are currently just concluding at UVRI CRP. And then the students' work, well, it's her doctoral work which she completed, where she was using in silico methods to find new products for malaria prevention. And that's it. Thank you.
Okay, we really appreciate your presentation, Brenda, and excited to see what work you're doing in Uganda around schistosomiasis and malaria and the vaccine space. Any questions from the floor? I know it's lunchtime, but at least one of you. Okay, Brenda, maybe you can tell us a bit about, you mentioned the population you're looking at possibly co-infected. So when you're doing some of your malaria studies, did you look at the impact of schistosomiasis against the malaria vaccines that you were testing, or they were
Thank you so much, Lucy, for that question. Well, for the malaria epi study, it's really just basic. We want to understand the prevalence of malaria. We are not yet looking at a product. That's in future. So we haven't looked at schisto in this case. However, for some of our previous studies, we were able to look at co-infection. We had a study which was a simulated HIV vaccine study, but of course, as you know, there is no HIV vaccine. So the stimulation was actually with hepatitis vaccine, and we were able to show indeed that those who were infected with schisto had lower responses to the hepatitis vaccine. And as I mentioned, we also have a published study which was looking at responses to yellow fever vaccine within the same population. So we already know that presence of these worm infections do modulate the immune responses. But in terms of what we are doing in preparation for the malaria vaccine, we were not looking at responses. What we are trying to understand is the prevalence of malaria, because the vaccine is looking at a higher age group than what the vaccines currently rolled out are looking at. Okay. Thank you.
All right, so with that, Brenda, we want to appreciate you once again. Let's just give a round of applause. Thank you very much. Brenda, in case anyone wants to reach out to her, I think her email will be available and we're happy to share. Okay, so thank you. So now we're going to move back into the room. We've got 3 more presentations. I would request the speakers who are remaining. Unfortunately, when you're on the floor, I can afford to be a bit harsh with you. I'm only going to give you about 10 minutes because we want to ensure that we come back in time for the presentation at 1:30, which is online. So let me allow my colleague, Dr. Rebecca Ayako, to come to just prepare herself. In terms of her bio, Rebecca started her master's degree at Kenyatta University, and she actually came to Kipre to do her master's project on Leishmania and Plasmodium falciparum, Plasmodium braggi. And she did a study in mice. She then went on to do a PhD at the university, at Moi University. And there she was looking at vaccine responses in the context of COVID. She's now back with us as Kipre, as a full-time research scientist, where she's looking at Leishmania and which other diseases?
Co-infections.
Just generally co-infections. So Becky, it's really nice to have you on board. We look forward to your presentation, and please keep to time.
Yes.
Thank you so much.
Thank you. Okay, so thank you. One of Kipre's mandate is to translate research into actionable strategies. And in our Leishmaniasis lab, we're trying to translate immunological research into guiding Leishmaniasis control. So I'm going to give a brief overview of the milestones we've done and the current projects and our future direction. So in a snapshot of introduction, so 1 billion people are at risk of contracting Leishmaniasis, and there are approximately 1.5 to 2 million cases reported globally. It's endemic in 98 countries. We see parts of East Africa, Southeast Asia, and Latin America, with annual deaths of approximately 20 to 30,000 deaths. So the clinical forms of Leishmaniasis, so there are 3 major clinical forms. We have the visceral, that's VL. It results to hepatosplenomegaly, that's you can see in A. And it's caused by Leishmania donovani and infantum. Secondly, we have cutaneous Leishmaniasis. It forms ulcerations in the skin caused by Leishmania major and other species. Thirdly, we have mucocutaneous. So we can see this individual has maculopapular lesions in the nose and mouth. Sometimes it can go to the throat. And lastly, this is post-Kalaza dermal Leishmaniasis. It's a complicated form of VL. It normally reoccurs after treatment from VL. So a brief overview of the immunological architecture that guides us in terms of coming up with vaccines and treatments. So when one is bitten by infected sandfly, the promastigotes, that's the infective stage, is taken up by macrophages or the dendritic cells, which are the professional APCs. So this triggers Th1 responses, resulting in secretion of pro-inflammatory cytokines, which we can see there's IL-2, IL-12, interferon gamma, and TNF-alpha. So that's protective in terms of reducing parasite burden. On the other hand, when we have high pro-inflammatory cytokines, we result in a switch that's called Th1, Th2 switch to Th2 responses, where we have high IL-4, high IL-10, and TGF-beta. So at Kipre, one of our major pioneers is coming up with animal models, where we are able to do vaccine development in terms of coming up with adjuvants, co-infection studies, and also natural product immunotherapy. So let's start with the animal model. So Olobo in 1987 with his colleagues were able to develop this vervet monkey. Scientific name is Chlorocebus aethiops, or common name African vervets. So they were able to develop it and validated it in 1995 by Gisheru. So this model is able to develop the clinical manifestations of both cutaneous and visceral disease. So in terms of human VL, it is able to mimic it in terms of immune responses, focusing on T-cell activation patterns, antibody profiles, and cytokine dynamics. So our second mandate is to come up with vaccine development, so we are focusing more on adjuvants. So we did a study on alum and BCG, comparing it with MPL, which is a toll-like receptor 4 agonist, which is able to promote Th1 responses, which, like I said earlier, it promotes parasite clearance. And thirdly, we tried using MISA, which is an oil-based adjuvant, which enhances T-cell priming. So we found out that in A, when you use alum and MISA, it was able to promote Th1 responses marked by interferon gamma. And in B, the parasite burden in alum and MISA was quite low, which is actually protective. And in C, when we're comparing the Th2 responses, it was quite reduced. So when Th2 responses reduce, we improve Th1 responses. And also we tried to measure antibody responses. So we had the isotypes IgG1, 2, 3, and 4. So IgG1, 3, and 4, they're not protective, but they're a marker of disease progression. So we saw that IgG2, which is protective against leishmania, it was quite high in alum and BCG. So this makes them primary candidates of adjuvants when they are combined with Leishmania donovani antigen as the sonicate. So thirdly, we are doing some co-infection studies. So we did Leishmania donovani and Plasmodium berghei in mice. Also Leishmania major for cutaneous leishmaniasis and Plasmodium berghei. So in figure A, you can see those are the malaria parasites. We have different stages, rings, schizons, and trophozoites. In figure B, we can see those are, it's a splenic impression smear, where these are the splenocytes infected with the mastigotes. So you can see some of the parasites are like rupturing the splenocyte. So in both cases, we have the early stage suppression where for both parasites, the parasite burden was quite low in the first stage of infection. So this is linked by Plasmodium berghei inducing Th1 responses, which is able to downregulate or reduce the parasite burden for both. But as Th1 responses increases, it's like a switch in the body it's an immunomodulatory mechanism where it switches now to Th2. So that's why we have the immune exhaustion phase. So that favors the parasites, so they're able to multiply, leading to clinical implications such as severe histopathology and high mortality rates as observed in that study. So also, we tried to do some Leishmania and Shiv co-infection studies. So currently, we developed a Shiv model to study both the immune dysregulation and identify those checkpoint exhaustion markers. So this study is currently underway, so I don't have any preliminary data. So this, the virus, we used SHIV because HIV will manifest what happens in the immune system of a human, and SIV has the receptors so that we were able to now infect the animal. So lastly, we have current studies going on in our lab. We're trying to look at both macrophage polarization, where the M1 switches to M2. So M1 works the same as pro-inflammatory cytokines. So if we understand how long the M1 is sustained, and when it switches to M2, we are able to have or come up with vaccine strategies and also use of monoclonal antibodies like the ones they use in prostate cancer to reduce the parasite burden. Also in terms of co-infection modeling, we're trying to look at more co-infections in terms of TB and Leishmania, and also in SHIV, focusing on other, expanding the study. In terms of immunomodulatory mechanisms, we are trying to look at plant-based studies, plant-based products as adjuvants which are able to enhance Th1 responses. And lastly, like from my title, it's from bench to field. Most of this work is bench work, and in terms of field work, we are trying to integrate genomic profiling with cytokine signatures to identify predictive markers, because some of the individuals in the field, although they get bitten by infected sandfly, they don't develop an actual disease. And that could help us to understand why these, some individuals are protected while others develop severe disease. And also in the lab, we're trying to come up with more vaccine efficacy research and personalized treatment responses. So I think the 10 minutes is okay. So I'm grateful for everyone.
Super, thank you for keeping time. As Sarah's moving up to set up, allow me to see if there are any questions. Leishmania, malaria? Yes.
Hello, my name is Huma. So thank you for your presentation. What can you say about T-cell exhaustion as a challenge in immunotherapy development of immunotherapies, especially against leishmaniasis? You've mentioned about MLP as a Th1 agonist. In this case, there is immunomodulation. Th2, there is a favorism to aura bias to Th1. So after treatment or parasite clearance and disease elevation, how do you make sure that there is a Th1, Th2 homeostatic state? How do you revert that to homeostasis?
Okay, thank you. So in terms of T-cell exhaustion, so those are two questions. So in terms of T-cell exhaustion, so we have like T-cell exhaustion markers, that's CTLA4, PD1, PDL1. So if we use monoclonal antibodies that are able to block those receptors for T-cell exhaustions, we are able to promote Th1 responses. But in terms of our genetic makeup or how we are made, there's a balance. So as much as we move to Th1, high Th2 responses, it will continue to shift back to Th1 until there's normal homeostasis. Okay.
Okay, thank you. So Becky is available, so if you have any more questions, kindly you will see her during the lunch break. So let's load the next presentation kindly. So allow me to introduce Dr. Sarah Kitui. She's a biochemist and a lecturer and researcher with extensive expertise in molecular biology, immunology, and microbiology. She's a recipient of the HBNU Fogarty Global Health Fellowship from 2022 to 2025. Congratulations for that. Awarded by NIH in collaboration with Harvard University, the T.H. Chan School of Public Health. She gained her PhD in biochemistry from Rhodes University in South Africa, and also has an MSc in biotechnology from KU, Kenyatta University. Her research is driven by a focus on protein-protein interactions in both hosts and vectors, and the influence on disease progression, particularly in malaria and cancer. She has contributed significantly to the understanding of the role of heat shock proteins and their co-chaperones in disease biology with translational implications for biomarker discovery, therapeutic targets, and vaccine development. So we welcome you, and please keep time. Thank you. I won't pressurize you, don't worry.
Thank you so much, Lucy. I really appreciate it. Ladies and gentlemen, allow me to share with you our work on the immunological insights into upregulated Plasmodium falciparum chaperones and surface antigens in pediatric severe malaria anemia with implications for vaccine development. Great, so a lot has already been said about the global burden of malaria, especially the heavy implications in the African continent. But I wish to re-emphasize the fact that within this African continent, the largest proportion of malaria cases present in young children, and in regions like Western Kenya, where this study was conducted, severe malaria largely presents in the form of severe malaria anemia, which is characterized by hemoglobin levels of less than 6 grams per deciliter of blood. So our study in particular focuses on understanding the immunological and molecular determinants that drive severe malaria anemia, with special interest on the role of molecular chaperones and their cooperativity with Plasmodium falciparum surface antigens. So the molecular chaperones in themselves collectively refer to a group of proteins that are normally utilized by eukaryotic organisms, including the Plasmodium parasite, to foster what is collectively referred to as protein homeostasis, or rather protein folding. And the heat shock proteins, designated up there as HSPs, are a unique group of the molecular chaperones that are normally utilized whenever eukaryotic organisms are subjected to stress. And in the HSP protein folding machinery, as illustrated in the far left, it's not going back, okay. Great. So in that protein folding machinery, the protein that needs to be folded is normally recruited into the protein folding machinery by members of the early protein complex, and then trafficked into an intermediate complex where, with the help of the cooperativity between different members of the HSP machinery, it's subsequently sent into the lead complex. And the end product of this machinery is normally a release of a protein in its nicely folded or stabilized state. Now, the Plasmodium parasite has been shown to dedicate a big chunk of its genome towards the expression of different HSPs that are normally utilized during different forms of stress, such as hypoxia and thermal stress. And in particular, it's been shown that when the parasites transient the temperature changes from the mosquito vector into the human host, that transfer is normally associated with changes in temperature that require stabilization, and that stabilization is offered by the role of the HSPs. It's also been demonstrated that within the human host, especially during febrile illness, when there is elevation in temperature, the Plasmodium proteins are normally susceptible to degradation, and the HSPs come in handy to ensure that they are not degraded. And beyond stabilizing and ensuring that other proteins are nicely folded, these HSPs in Plasmodium falciparum have been shown to also influence the protein-protein interactions, particularly the interactions that occur between the Plasmodium proteins and the human host cells. So over the years, there's been some sufficient evidence from different labs across the globe indicating the utility of these HSPs in as far as immunogenic roles are concerned, and certain HSPs like HSP70 and HSP40 have been demonstrated to have the capacity to stabilize components of virulence, especially in Plasmodium falciparum. And therefore, guided by this background, we hypothesize that in the wake of severe malaria anemia, there is a cooperativity between these HSPs and the Plasmodium surface antigens, and this cooperativity ensures virulence and, of course, immune evasion. And we think that some of the implicated HSPs and the surface antigens could then be explored as vaccine candidates specifically for severe malaria, so that then we boost the repertoire of the readily available vaccine candidates illustrated on the far left. And therefore, guided by this background, sorry, this thing is just not moving, and that eats into my time. Okay, great. So we then sought to test our hypothesis using these aims. So we basically defined the differential expression of a couple of these Plasmodium falciparum chaperones, including the HSPs, and of course, a couple of those Plasmodium falciparum surface antigens. We also identified a couple of the targets that could then be evaluated as biomarkers and vaccine candidates. Not sure if, okay, great. So for us to have achieved our aims, we conducted a comparative study where we basically used RNA sequencing on whole blood samples collected from two groups of children, one presenting with severe malaria and the other presenting with non-severe malaria at Siaya County Referral Hospital within Western Kenya. And we proceeded to perform differential gene expression analysis where we identified a couple of HSPs, and we also subsequently used a couple of commercially available antibodies to test the ability to detect Plasmodium-specific HSPs. So this table here illustrates the characteristics of our study participants. These were largely children between the ages of 0 to 36 years. And what was quite striking is that the ages of the study participants in the two groups were similar, which was quite good to safeguard against age-related biases. But what we observed in relation to the hematological parameters was that the hemoglobin levels alongside the hematocrit levels and the levels of red blood cells were significantly lower in the group with severe malaria anemia relative to the group with non-severe malaria, which is consistent with what is expected for the clinical definition of severe malaria. In addition to that, we also observed other hematological parameters, like the mean corpuscular volume was way higher in the group with severe malaria anemia compared to the group with non-severe malaria, and that is also in agreement with what is expected clinically for severe malaria. So collectively, we think that these characteristics aligns with the correct definition of severe malaria, and therefore, in this regard, our grouping was correct. It's the thing not moving, not me. Yes, please. Okay, sorry. So the volcano plot on the far end just illustrates the distribution of the Plasmodium falciparum mRNA genes. And we had about 499 genes that were upregulated in the group with severe malaria. And the same group, we had about 67 genes that were downregulated. And this table here illustrated using the transcript abundance in addition to the log2 fold change at a p-value of less than 0.05 did depict an upregulation of a couple of HSPs, including HSP 40, 60, and 70 in the group with severe malaria relative to the group with non-severe malaria. We also observed a concomitant increase in the expression of a couple of Plasmodium surface antigens, like the entraps, in the group with severe malaria relative to the group with non-severe malaria. And where did you say I point? Yes, please? Okay, we're here. So this here just illustrates the trend of the distribution of the upregulated HSPs and the Plasmodium surface antigens. And what you see here, which is what looks like a trend where the Plasmodium antigens have a higher magnitude of the upregulation compared to the upregulation of the HSPs is very much expected in the wake of the fact that the HSPs in themselves pass as molecular chaperones that are meant to serve as catalytic helpers to potentiate the expression of the virulence factors on this end. And therefore, in that regard, they're not expected to be upregulated in the same magnitude like the plasmodium antigens. But it was quite nice to see the upregulation of unique candidates like NTROM4, which, unlike PFEMP1, which has been a challenging candidate in the wake of vaccine design owing to the fact that it's quite polymorphic and it's also species-specific, NTROM4 is a unique candidate because it is expressed by multiple Plasmodium parasites. It is also very conserved, and unlike PFEMP1, it's also expressed in the early phases of the inversion of the red blood cells. So that puts it as a fair candidate in comparison to PFEMP1. In the wake of the HSPs, we appreciate that they are on their own immunodominant molecules, and therefore they could be used as standalone vaccine candidates. But we also appreciate the fact that they serve as molecular chaperones, and therefore they could be used as molecular adjuvants to potentiate the delivery of the Plasmodium surface antigens on this end. So we also use some commercially available antibodies to test the expression of the Plasmodium HSPs in both Plasmodium culture lysates and infected patient lysates. And we did observe a nice expression of the HSPs in both the samples, the lysates from the cultures, and of course the lysates from the infected red blood cells, which in a way agreed with what we see at the transcript level. So in conclusion, we did observe that indeed the exportable Plasmodium HSPs are upregulated in children with severe malaria anemia. And we believe the data underscores the role of HSPs as both potential biomarkers and immunodominant targets that could be explored for next generation vaccines. And this is largely informed by the fact that these HSPs have been shown to stabilize virulent antigens like PFMP1. So moving forward, we are hoping to examine the co-localization of these HSPs with some of the Plasmodium surface antigens like PFEMP1 and the NTRAPs by immunofluorescent and proteomic analysis, and we'll also be assessing whether these HSPs and the antigens can form complexes that can indeed be used as molecular adjuvants to enhance vaccine efficacy. So I'm highly grateful to especially those study participants and their families and the clinical and research teams in both Sioya and Kisumu where the data collection was done. At the time of these studies, I was a Fogarty Fellow, and I'm grateful to the NIH Fogarty Fellowship for funding my fellowship, and to my mentors, Professor Douglas J. Perkins and Professor Collins Ouma, who's seated here with us alongside Dr. Sam Anyona of Maseno University for the mentorship, and my current host lab, the Gitaka Lab in Mount Kenya, where I currently serve as a postdoctoral fellow. And thanks to you for giving me your attention in the work of the challenges.
Thank you very much, Sarah. I know I've taken over a role that you guys haven't seen me in before. So thank you so much, Sarah, for your good presentation. Anyone with questions? Just one question because of time. Anybody? Yes, please, Lucy.
The heat shock proteins and the relation to, I mean, if you have like another infection, bacteria, etc., would you not be concerned that they would be upregulated because of the other diseases that the person might have, or is not a problem? Are they specific? Like the ones you mentioned, the HSP 110, specific just for malaria, or you can have it also upregulated with other infections?
They are ubiquitous proteins, so they are expressed in different, I think, scenarios. But in particular, what we have here are Plasmodium-specific HSPs, confirmed at a transcript level and a protein level. So there is not any case of non-specific detection.
Wait, sorry. Tom Hope, Northwestern University, and what's your feeling about the fact that everybody is talking about stress proteins in almost all these talks?
Well, there is a lot of the role of stress in disease biology, and especially where pathogens and just how they interact with the host is concerned. And I believe the stress response can actually be explored for value in the work of the design of cutting-edge technologies for both infectious and non-infectious infections.
I believe that was a beautiful answer. Thank you.
It's a great honor. Thank you so much.
Thank you so much, Sarah. Please, let's give her a round of applause. Okay, I'd like to quickly introduce our next presenter. This is Dr. Bartholomew Ondigo from Egerton University. He has trained from Maseno University and the US, and he's a current lecturer at Egerton University. He studies malaria immunology in Western Kenya, and he's passionate about training students in class and in biomedical research. So please let us give him an opportunity to take us through his presentation. Karibu, Doc.
Thank you, Winnie, and thank you, the presenters, for giving me time to present my work. And thank you for all of you for finding time to be here, despite being just a step to lunch. Please project my presentation, please. Yeah, that's my name. I have a small lab at Egerton University. We are trying to set it up. And what I want to present today is looking at levels of those two biomarkers in relationship to other inflammatory mediators, with a focus on women infected with malaria in Western Kenya. I'm based at the Department of Biochemistry and Molecular Biology at Egerton University, and this work is work in progress, so I really do not have very strong conclusions since it's something that is still ongoing. So to start with, just to ride on what Faith and Bernard had in mind, that malaria is a major global problem affecting majorly sub-Saharan Africa. And pregnancy malaria that was earlier studied by Mikhail Fry, Duffy, and Rogerson, we see these parasites sequester in the placenta, and we have molecules like CSA, VAR2SA that bind and make the parasites to sequester there. And as such, it causes maternal anemia, adverse pregnancy outcomes, preterm delivery, women deliver before the 37 weeks of delivery, low birth weight, less than 2.5 kgs of baby weight, stillbirths, neonatal mortality, and the worst affected are pregnant women and children, especially in Africa. And on this far side is a diagram of a placenta, and you would see the baby abortion, stillbirth, etc. So as part of the introduction, when these cells, majorly the monocytes, macrophages, sequester in the placenta, they form a kind of inflammation. And in our lab, our interest is to look at these monocyte macrophages and what their role is in placental malaria. And these monocyte macrophages, they do express two particular molecules. They do express CD163 and CD14. And when we have inflammation, the CD163 can degranulate, they shed off and goes into circulation. And the same thing, soluble CD14 is shed off and goes into circulation. So we are trying to really understand how do these two molecules, these two biomarkers, how are they relating with these other inflammatory mediators? As we do this, there's this whole issue of M1, M2, that M1 is pro-inflammatory, M2 is anti-inflammatory, M1 is more of destructive type, M2 is more of repairing type. So we are trying to elucidate, trying to shed more of the pathogenesis of placental malaria. And there are sets of markers that are used to they used to distinguish these cell types from M2A, 2B, 2C, 2D, and our work is to phenotype these markers in relation to infection status, in relation to gravidity, and in relation to the past parasitemia levels in these women. So we had two major aims. One is to profile fetal macrophages in the placenta of these infected and uninfected women. And two is to look at these levels of these activation markers in plasmas of these women. And our hypothesis is based on that when we have placental malaria, there are changes in these fetal macrophages, either from M1 to M2, that impacts on these biomarkers and ultimately cytokines, and ultimately we have adverse outcomes on the fetus. For this presentation, I'll focus on objective number 2. Objective number 1 is still we're still analyzing the data sets. So this was our study design. We enrolled women at around week 28 there. At week 32 of pregnancy, we collected peripheral blood. At delivery, we collected placental blood. We collected the placenta for flow cytometry. Placental blood was used for soluble CD14, soluble CD163 ELISAs. And microscopy was done for determination of whether they were infected or uninfected. We also had data on their demographics and socioeconomic status. And for this study, we had a sample size of pre-gravid women. These are first-time mothers, 125, and multigravid mothers giving birth to the second, third, fourth, there were 234 in terms of the sample size. So what we see here, at gestational week 30 and 32, for soluble CD14, when we compared preemie mothers compared to preemie that were infected versus uninfected, we see that infection results in increase of the soluble CD14, and this was not seen for the multigravid women. And for this soluble CD14, multigravid women had elevated levels of soluble CD163, but that was not seen in the preemie-gravid mothers. When you looked at the placental blood, more or less a similar picture was seen, that infection in the preemie-gravid results in an increase in soluble CD14 and results in an increase in soluble CD163 in preemie-gravid BS-positive mothers. We moved ahead. We tried to relate what these biomarkers with established either Th1 or Th2 cytokines, and what we showed is that soluble CD163 correlated closely with IL-10, that is at gestational week 30 to 32, and at delivery we had IL-1 beta, CXCL9, and TNF-alpha correlating closely with soluble CD163 levels. However, the correlations were not seen for soluble CD14 levels. In summary, what we are trying to say is that in women who are infected with placenta malaria, at gestational week 30 to 32, they have elevated levels of soluble CD14, soluble CD163 in peripheral blood. This upregulation is mostly seen among placenta malaria positive preemie-gravid women, and the placenta soluble CD163 levels correlated with CXCL9 and TNF-alpha, which probably also informs why we have more inflammation among these preemie-gravid women, and probably why kids born of preemie-gravid women tend to be more like low birth weight and preterm in delivery. All this suggests that there is continual chronic inflammation in the placenta, and the high levels of soluble CD163 correlated with IL-10 in primigravid women. And the same thing here is that when we see high levels of IL-10, studies have shown that IL-10 is related to preterm delivery. So both soluble CD163, soluble CD14 propagates or are involved in the pathogenesis of placental malaria. As part of acknowledgements, I work with students. I want to thank Duncan Wakoli, Dixon, George, and Evelyn, who are my master students working in the field, the study participants, and this work is done in the 2 hospitals in Busia, Port Victoria Hospital and Seaport, and support for this work is Bill Melinda Gates Foundation, African Academy of Sciences, and my host institution, Egerton University. And to you, thank you for listening to this presentation.
Thank you very much, Doc. Thank you for that presentation on inflammatory responses in placental malaria. So do we have any reactions? A question, just one, because we are behind in time. Any reactions, any questions? Okay, if there's none right now, I know. Okay, please.
Thank you very much for that wonderful Thank you very much. My name is Patrick Maura. I'm just concerned that you are basing your argument on correlations. And we know that correlations are not causations, right? So could there be other issues that could lead to the same sort of correlation that we could be concerned about?
Thank you, Pat. Thank you, Maura. Yes, for these preliminary findings, I'm basing it on correlations, but this is not the whole story. The whole story also has to involve phenotyping that we are currently doing with about 25 of these placental tissues. And so when we relate the phenotypes, what markers are they expressing, the infection status, and these biomarkers, then the story will be complete. But I appreciate your concern.
Okay, if we have any other concerns, please find Dr. Ondigo during lunch break. Thank you so much, Daktari. Please, let's give him a round of applause to say thank you. I will hand over back to Dr. Lucy to give us the way forward for lunch.
Okay, so really, I want to appreciate all the speakers for this session. It was a very stimulating conversation, predominantly around malaria, but I think that still is a problem for us here in Kenya and within the continent. So I want to just make a few announcements kindly before we break for lunch. The first one is you've hopefully all been given these sort of vouchers. If you haven't, there'll be a lady, Joy, if you can just raise your hand. Joy over there has got the meal vouchers. The green one is for the 10th floor, and the brown one is for the the 3rd floor. The food is the same, don't panic. It's just that we're many, and we have to spread between the 2 floors. We'd also kindly ask that you mount your posters at lunchtime. There'll be an evaluation later in the day with the hope to give you a prize at the end of it, so please just give it your best. And in case you want to know where, for day 1, please refer to the abstract book so you know if you're here, you're presenting today your posters or tomorrow. Okay, so we're at hand if you want to ask any questions. We are breaking now, and we appreciate we are behind time. About 30 minutes, but please, if you can come back by 2 o'clock, we'd appreciate that. Thank you and enjoy your lunch.